learning. Like I kind of have bits and pieces of your background that you've told me in
Third
the
thing I
past,
would like
but
to say, it's completely
tell me your
unethical
life
on the
story.
grounds that companies do
I don't
that
usually ask that, but I really want to hear.
Financial basis.
How
Like,
much time
okay,
do
this
we
woman is
have? Like,
dedicated to her
version,
I should cut you down, but like,
okay, this woman doesn't really seem dedicated.
but how did you get into
so
tech in the
going to
first
be a
place? I
candidate
guess.
who's going to probably
So
take maternity leave.
it's
I don't
it's
really
a funny
think we should hire
story
this. I don't
because
really think we should.
I come from
It's not.
a very
It's going to be a
non-tech
completely shadow
background, as
benefit based
fuzzy as you
system.
can think of.
It's going to lead to a
So
lot of
after high school,
discriminationary hiring. And
I
on
didn't
these
go to
three
college
grounds,
and I
I
started
think it's
traveling.
completely
Like
unethical.
the food pictures.
However
they don't
innovative a
like, that's what's
solution may be,
going on.
however nascent and
So
brilliant a
I did that
technology might be,
for 3
if it's
years,
not
and
for
in the
everyone,
process, I was
it's not
writing.
ethical.
I was writing for newspaper. I hosted a couple of columns,
Thank you.
and I wrote a couple of books, which
Yeah.
got me into
So
more
congratulations,
trouble
both
than
teams. You are
I wanted,
debating
than I
really
wished
so
for.
well.
Wait,
And
really?
I'm not
Okay.
taking any side, and I'm not giving my
Yeah,
personal
so,
opinion, but I just
like
want to
internet
ask team
popularity is
three.
like a double-edged sword. So
Mhm.
my books
See,
got
who
very
is
popular,
getting
and
delayed pregnancy.
okay, so it's like very popular stuff.
Usually,
Arrogant. It's
those
like
who are from lower
more
socioeconomic
popular
class,
than
they
I
get
was
they were
married later.
bestsellers, right? I think it's pretty fair to
Which
say that they were very
chances
popular.
they
What were the
In-
books
in
about?
Vietnam.
Doctors
So it's
and
about
IITs
traveling,
and
like
then
the people I met
PhDs
on
and
the
all.
road,
They
and
keep
I
studying
was
till the age
young.
of
I didn't know
30,
how to handle
32,
one's
then
attention,
fellowships and all. And then they,
people were like, "Oh,
get
it's
married.
like it's
So it is
impossible.
their
A girl
question
can't
basically.
possibly travel by
So
herself." And they were
what do
like,
you
"Oh,
think?
she
They
didn't
should
write
not
the
be
book.
allowed this
She
particular
didn't
thing,
write
because
any
if
of
you
that."
know the
Like,
age of
she had people like some
what
like
age
running
which is
things for her,
decided,
doing
that
things
after
for
the
her.
age of
Like, people
39 or
accused her of
40,
having a lot of money.
the quality of egg is not so good.
So there was a lot
And the
of
chances of getting
controversies, and
abnormal child
I
increases
was a little bit
enormously.
like offended.
So
It was
this
like,
particular lady,
who is
late
these people?
30s, let
Why
us
is
say
this like making me
36,
answer all
37
like these
each,
stupid questions?
should not be allowed. You
But
see,
at that time, I
you
did
feel
not know
so
how to handle that,
just
and
for the
it caused a lot of like backlash.
equality to this
So I
this
was
lady,
like,
just because
okay, I'm
she's
so
getting
tired of
married
this.
late
I'm going back to
for
school.
whatever
So I
reason. So
went back
it
to
should not be allowed.
Stanford.
You're saying. So what is the
And I
idea
wasn't
behind
even
this?
doing
What is
something
the
like
logic
writing,
behind
like
this?
writing or
Everybody should be having the freedom
like political
of
science.
choice. So
But then,
the woman,
do you know, I
single
was at
woman in
Stanford,
late
and
30s,
and people
should be
told me
given
that
the
the question
choice.
is not
Everybody
whether
should be having
you
the
should take
freedom.
a CS course or
So don't you
when
think so?
when you're gonna do it, because 90% of like Stanford
Thank
undergrad
you.
take
That's a
CS
brilliant
courses
question,
in
and
4
that's what my
So I
father
just took
also
a
brought
course
about
in
when
the
I
first
was
quarter,
debating this
and
with
I
him.
realized it.
And he
In your
just
first
told me
quarter
one
there?
thing, that you know what, I was
Wow.
born
Like,
to
it's
do
an
my
introductory
graduation
course.
studies there.
You came to Stanford.
Really
How old were
ahead in
you?
a career.
You had already been a
And I
best-selling
got
Ask
author.
that question.
Exactly. And what I'd like to say to that is pregnancy,
So I was like older than
delivery,
my classmates.
and raising a child will
and
never fit into
so I took my
any
first
woman's
quarter.
career
It was
plan ever.
fun.
No time is an ideal time,
And I
sir.
kept on doing more
Our
courses.
focus shouldn't
And
be
before I
on
knew
making
it,
it
it was a
easy
CS
or making
major.
it difficult.
And
Our focus should be on enabling the woman
I
to
took an AI
combine
course.
work
I
and
cried
family.
a lot the
We
first
should
class. It
help
was so
and support
difficult.
in every way possible with our social
But it
structure
was so very
to
flashy. I
fund
think I came
and
to
make
Stanford
it
at the
possible
time. It
that
was
it's
a peak
okay
of
for
the
her to
AI
be an
hype.
IITian, to
Uh-huh.
Am I
do
allowed to
or
say
to
AI
be a
hype
junior
here, or
resident
Sure,
is
in,
yeah.
it too
say,
touchy?
extremely
Okay.
difficult department of medicine, say surgery.
You can say literally whatever you
really,
want.
really takes time and still have a baby.
Okay.
We should not.
So
We
so
should
I did
change
that,
the
and
social
it was
outlook in
fun.
such a way that we don't look down
And yeah,
upon
here I
her,
am.
or we don't look
And I
down
think in my
upon a
third
junior
year,
or
I
senior
taught a
resident who's
course.
having
I
a baby.
created and taught a course.
And
Your third year's an
we're not
undergrad.
pushing for early
Yeah.
motherhood. We're pushing for
Wow, I try to think about what I was doing as an undergrad, and I feel I feel embarrassed.
Problems if,
Okay, to be fair, I was like older
complications if
than
they
people,
indulge in
right?
this.
I
And you
also
can
had
only go so
I also
much
didn't
against
have
the
to
laws of
spend
nature.
time on like frat parties,
At the
like
end, you really
impress
need to
people.
realize there's a
I was
world.
pretty much
There's
done with the
a
party
point
scene
where
by
we need
then.
to stop.
Right.
Itna
And so
bas.
you
Like, we
but you
need to
taught a
understand
really
that
popular
इसके
class, right?
beyond it's a damage to your
I
own
think it's
self,
you
it's
could
a
say
damage
it
to
was a
the
popular
baby
class.
itself.
I could
It's a
say.
damage to the baby's
I think
life.
people can say
The
that. I
baby's
feel
going
like
to have
I
a
feel
lot
like
of genetic
it was
abnormalities, chromosomal aneuploidy,
it was
God knows what
unexpected.
not.
Like I
And
didn't
there
even
has not
know
been
that the
enough
course was
studies about
popular.
it.
I was just
This
like
technology is
ditching
very
it.
recent.
And
It's
one day I walked into
2012
dining hall,
onwards.
and a friend
Like,
was
we
like,
don't have long-term
"Did you see
data.
my
इसको हम
comment by you
public
on
policy
Hacker News?"
policy
And I
में
was like,
कैसे
"Why
डाल
would
सकते हैं?
anyone say
I'm
this on
sorry to
Hacker News?"
interrupt here. We don't
And
have
it
much
turned out
time
that
with
my
this
course
right
on the
now.
front page of like Hacker News.
Can we
I'm like,
move
wow,
to the
it's
next
interesting. And at some point,
question for the
Yes.
next?
yeah, I
the
never know what
proposition
happened.
team?
I was not like really active on Twitter back then. And it's just one day open Twitter, I have like 10,000 followers. I'm like, wow, who are these people? It was great.
Well, it's a timely class. Like, what was the topic of the class?
Hello,
Just
first team,
for people
team
might not know.
Oh, it was
Hello?
TensorFlow. I think
Can you
I
hear
think
me?
it was the right
Yes, sir,
time.
yes,
Like
sir.
TensorFlow was very
So,
popular back in
see, there's a cost
2016.
involved in this particular
Wow.
procedure.
It's
If you
crazy
see
how
even
fast
the
things change.
IVF
Like back then,
also is a
2016,
very
like
costly
dental
thing.
was
All
all
these
what we
procedures
talk
are very
about, and
costly.
now like it's all what
And these
people talk
procedures,
about.
as per the government policy, are
Yeah.
usually not available
So
in
a total
most of the
cost
government
on
hospitals and
TensorFlow, I
medical
think the
colleges
official
attached to
name
government
is
hospitals. So
TensorFlow for
all
Deep
these
Learning Research,
mothers,
which is also a
single
very
mother, you will
flashy
say,
name.
expecting, will have to go to a private
And
setup.
so I think I
So
put
don't
a
you
lot like
think
all
this
the
will
materials
promote
online,
commercial
and I
aspect
think it was
more
maybe the
than
first college level course
social
on
or
TensorFlow.
ethical or scientific
And
aspect?
also like
2016,
the
I'm trying to remember it. Like, I think you had to compile it
particular issue.
yourself, right, to get it to use the GPU back then. I'm trying to remember. I remember just installing
So
TensorFlow was a
firstly,
pretty painful experience for me.
we want to talk
it was
about, we
not.
want to
I don't
elaborate
remember
more
it to be
about
so
on the
painful. It was
autonomy
just
of
some
the woman to go
concept
for
was
this
a little bit
procedure.
hard to grasp,
And
as in
it
like
is
a
but
computation
in her
graph.
benefit
So you have to like build a
to
graph
delay
first
her
before you can run
fertility
it.
if she wants to.
So I think with
Uh,
TensorFlow 2.0 right now
and
is a bit different.
Aishwarya, would you like to elaborate on
yeah.
the public and private
Got it.
partnerships that
And
we
so, I mean,
proposed?
how did you come up with the
Right, sir.
material for that
But
class? Like, how did you
public-private
think of,
partnership
like,
is
what
not
to-
So
happening
when
for
I
these reasons.
started teaching the course,
If
I
you
was
see so
just
many
hoping
other
to
aspects,
learn.
PPP
So as you know,
is
I was
working, but
like,
not
so I
in this
started
particular
thinking about the course as like
area of medicine.
sophomore summer.
Because
we begin-
government is not feeling
so
the
I didn't know a
priority,
lot.
not willing to
I
give
was
priority
at
to this particular
as
aspect,
a ML startup,
which is very
and I
important.
used TensorFlow
Motherhood
for that
is very
startup for
important.
the internship, and I
Right, sir.
and I
So
was
I believe
like
this
blown
is
away.
why
I
we're
was
having
like,
this debate
wow,
right now
I didn't
here,
realize
because
you could
since
do
it is
so many
available in
things with it.
commercial settings, but
So I
not in
went
a
to like a couple of
public-private
professors
partnership
and
or
was
in a
like,
government setting.
can you
It's
teach a
because there
course
is
on
not
it?
enough
And my
awareness,
professor
and
was
we
like,
believe
I
that
don't
by
have
making a
time. Like, why don't
policy,
you teach
it's
it?
not that tomorrow
So
everyone could go to
so
a
it was
clinic
okay.
and start availing this
And I
particular
got a lot of people to help
facility.
me.
But over
So I
time,
have
with
some
this kind
friends
of
of
demand, if
people
we create a
who know a
demand
lot
in
about
society that
TensorFlow.
okay, there are more and more women who
I
wish
have
to have this
professors
kind of
who
procedure,
like
it would also
overlook, like take a look, like
incentivize the government to have this
who
kind
look
of
at my
funding in.
curriculum.
So we also
and,
would like to bring about one more
and,
point, wherein
and
even if
also
there is
gave a lot of feedback.
commercialization,
I
it
read
produces
lecture
a lot of
notes.
revenue for the
I was
country,
really
because
nervous.
just like
So
plastic, just like India is a
I
plastic
had
surgery hub, it would then
so I
become
had
like
really
an
good
oocyte
friends
cryopreservation hub.
was
So in that
coerced
sense,
into
the
being my
country's
fake
economy would
students.
keep growing, and it
So
would
if
probably help us
every
find
like
funded
lecture,
for
I would
people from the lower
I
economic
would make them
status
sit
and so on.
and listen to
Can't
me
afford
give them
it.
like a
Right.
fake lecture.
Really?
It was yeah.
Okay, thank you so
Yeah,
much.
so I think I got a lot of help. So it was like learning.
Okay, so
together
now
with
we'll
my
sum up
students.
the
I didn't
debates
think
with
of
the
it
closing
teaching as much
speeches.
as
We'll start with
like
the
group
closing
study.
speech of the
That's super
proposition
cool.
B.
Did you?
So you can
when
introduce
you,
yourself and then
came to
we'll
Stanford,
begin.
you hadn't taken
So
any computer science
we'll
class
start.
before?
Okay.
So I came from a
So
math
hi, my name is
background.
Aishwarya.
So I
I'll
did
be
math
starting in 3,
in high
2,
school.
1.
So
In
I
this
think,
day and age, why is
I think
giving
like
women
we also took
reproductive
some
autonomy
CS
even
courses,
being
but you know,
debated?
like
Oh,
it's
yeah.
like
Why
more
does
like
the society
very
still
very
view
basic.
women as
I
baby
don't
making
remember, it was
machines
like
and shame
blue
them for not
screen
being
back
settled
then.
with a husband and 2.5 kids by their 30s?
Wow.
We, on side proposition,
Yeah.
completely reject
It
this
just seems really
paradigm and instead
amazing.
give
Like,
you a
you know,
feasible
you went from
policy that empowers women in the
introductory
truest
to
of
computer
senses.
science
A study by Messin et al.
stuff
found
to
maximum benefits
teaching
in
a computer science, you know,
cryopreservation was
teaching a
performed between 32
TensorFlow
to
class
37
like
years.
2 years
This
later
only
is like
substantiates a
amazing.
policy of offering
Do
social
you
exclusion
have
to women
any
in their late 30s.
thought, like any
Our
advice to other
policy
people
thus
that,
allows
that
women to
want
press
to
pause on the biological
learn this
clock,
stuff?
focus on their career prospects,
I think
introspect,
this
and
is all
right
the
time to
beauty
find the
of
right
computer
person to spend
science.
their
Is
lives
the
with.
entry
This is
barrier
further
is really
evidenced by
low.
yet another study by
So also
Hawkeye et al.,
ML,
which showed
and
that
also
83%
especially with the
participants who were
experiment
single at the time of egg
oriented
freezing, of them, 39% of them had
progress
found life
that you
partners when they were
actually
surveyed
like
5 years
don't
later.
need to know
They
a lot
also
of
protect women from
theories
making
to
irreversible,
make a
life-altering
contribution.
decisions under societal pressures.
So I've seen
This
people
claim
who
is
get
backed by a
into
cross-sectional
ML
survey
for like a
conducted by
year and
Hammerberg
is
et
able
al.,
to make
which showed that
like
6% or
great
only
project.
6% of the women
Which
who
I mean,
closed
I'm
their eggs
still like
returned to use
ambivalent
them.
about it.
The
Like,
vast
it's
majority did
good
not
as
wish
it
to be
like
single
lowers
parents
the
or
entry
any
barriers
Your
and
time
like
is up.
allows more people from
Another
different
important
backgrounds
factor
to
is the
join.
low prevalence
But so
of
like, what does
decision
it say
regret in
about
females who
Phil
have
when
undergone
like
OC, as
somebody
proved
who
by
like
Greenwood et al.
joined for a year
Frozen eggs will
can
remain
make a
frozen, may even
pretty
be used for some
mind-blowing
years. However, there is no
experiment?
return policy.
Sorry
So
to
I don't know.
interrupt, but the time is up.
I know
Maybe it
how
means
to
there's
feel
lots
about
of interesting stuff
Teachers
to
are also, while our
try.
policy currently caters to well-off, educated, financially stable women, we don't believe that once it gains momentum,
momentum, it will be reflected
Yeah,
into the
I
rural parts of
don't
the
know
country.
any
I'll
example
have
of
it all.
giving
Thank you.
advice.
Yeah,
Yes,
time is
yes.
up. Yes.
I think it's just
Okay,
like get
thank
your
you
hands
so
dirty,
much.
like try
Now we would be
try
moving
things
on to
out,
the closing
and
speech of the
befriend
opposition
smart
team.
people. I think that's all.
Befriend smart
That?
people.
I have friends smarter than you. I don't think I can have
3,
got
2,
anything done
1.
without my friends.
I
really.
love how the
Yeah.
proposition
That's so cool.
completely brings up
I think
this
that's good
as
advice.
a insurance policy.
Why did
as an
you choose to
insurance
go into
policy, as an
AI?
insurance
For
policy,
me, it
very
was just a
productive autonomy.
promise
Ladies
of
and
AI.
gentlemen, social executing is
This
not an
AI
insurance
help.
policy.
So I
If
come
you opt
from
for this in your late
a village
30s,
in
which the
Vietnam,
topic says,
and
you're
I
more likely to
traveled,
go through a
and
lot
over
of
the time I
discomfort,
realized
a
that
lot of risk, And a lot of
language
expense.
barriers can
Side
actually.
note,
It
Israel
could be great if you can
advocates it in
overcome
their
the
national
language
policy.
barrier. For
Israel
example,
does
like
not
the
sponsor it.
majority of human knowledge
And of all this,
online
you
actually
still
written
have
in English,
You know why?
and people who don't
Because the
speak English they
chances
can't really
that the egg
access that.
that
Like
has
people in my
retrieved that has
hometown
been
like
retrieved at
can't really
37 or
read
late
anything
50s,
I write in English,
resulting in a
or my
live birth is
parents
really low. It's
are
between 2
afraid of
to
visiting me in
14%.
the US
All
because
this
they
for what?
wouldn't be sure how to navigate the
The
airport
question we should
or
be asking is why
how
are
to get
women
here.
in this position?
So,
Where have
so I
we
think
failed as a
so. At this
society?
time, I was very
Because
interested in machine
if a possible
translation.
argument in the favor of
Reading,
social activism could be to avoid
yeah,
discrimination between men and
like
women,
if you can
we
automate,
wonder if
like
equality
an automate
between
the
men and women should be
translation
achieved by completely
process,
erasing the
then it could be
biological
Your
really,
differences.
time
really
is
helpful. Then if
over.
we can overcome the language barriers and help people from maybe my village can access
Thank you to all the
human knowledge
participants and the
or
judges. It was a
just to
great
step
debate to
out of
watch.
the
Now
border.
we'll be moving on to
That's
the
so cool.
next debate. So the
That's what I
participants are
thought back then.
requested to
Very
wait in the
idealistic.
waiting room,
Yeah.
and
What are the
we'll let
topics
the
that
next
are
debate
most interesting to you
participants.
right now in
Thank
machine
you.
learning?
So I think over time I think what we are lacking is better engineering in machine learning. So people are so there are two aspects, both as in engineering in research and engineering in production.
So we'll be
So
beginning with
in
the
research,
second
there are a
debate
lot
now.
of
The topic for this debate
researchers
is social media is
who are
crucial for
amazing
research
at what they
engagement.
do, but who
Speaking
also
for the
like are
motion, we
not
have
the great
team 2
engineers,
with
and
Anuj
it's not
Toshi and
because
Pranav
like
Bhatia.
of them. It's just
And speaking
because
against the motion, we have
human
team
like
9
our
with
time is
Sarthak
limited.
Pawar
We
and
focus too
Upasna
much
Saha.
on research.
So
We
let's
can't
start right
expect
now.
them to be
We
like
start
great
with the
engineers.
opening speech
So
of
I
the
wonder if there's a
proposition
better
team. So
is we
you can
can
give your
give you a good
introduction and start with the opening speech.
tool set to help researchers carry out the research
Hello,
more
respected judges
efficiently
and my
and
fellow
have like more.
participants. Good
And
evening.
also, if
Hope
like
you're
if you
doing
have
well.
clean code,
I'm Pranav
it's
Bhatia, a
easier to
second year
control
MBBS
experiments
student
and
from
like
St.
help
G.S.
with
Medical College and
reproducibility.
KM Hospital, Mumbai.
And in
Today
productions,
I'll be
I also
speaking
think
for
that
the topic
there are
that is social media
people.
is
So there's a
crucial
gap
for
between
research engagement.
researchers and production
I
engineers.
would like to start off
I
by
think there's a
saying that
lot of
all the
have been a lot of
participants in this
progresses
debate are
in
currently
research
pursuing
machine
their
learning,
studies at the
and now this is a
undergraduate level.
question like how do
Technically,
we
some of
bring
us
the
must have
research
invested
into
some of our
productions,
time in
and that's what I'm
conducting
very
research.
interested in.
We know
And
that the
also
basic
startups that I'm
essential
part of right now
prerequisite
is
for
also
conducting
focusing on
research
that,
is
by
that the
helping
study should be
companies
feasible,
productionize
in the
machine
sense that
learning
it should
research.
be cost effective as well,
Hi,
and there
we'd
should be
love
an
to
easy
take a
recruitment
moment
of
to tell you
participation.
guys about
So
Weights
with this
and
respect,
Biases.
I firmly
Weights and
believe that
Biases is a
today's
tool
social
that
media
helps
has
you
emerged
track and
as a
visualize
powerful
every detail
recruitment tool
of
to
your machine
disseminate
learning
the
models.
information which is
We help
related
you
to
debug
research.
your machine learning models in real
It is
time,
estimated that there are
collaborate
about
easily, and advance the
2.94
state of
billion
the art
active
in
users
machine
on
learning.
Facebook, and this has afforded
You
a
can integrate
remarkable
weights and
access
biases into your
to
models
not
with just
only
a few
the
lines of code.
usual population
With
but also the
hyperparameter
vulnerable
sweeps,
as well
you
as the
can
low
find
incidence
the best set
population.
of
It is
hyperparameters for your
noteworthy that it
models
is a
automatically.
Herculean task to
You can also
recruit
track and
adequate
compare
number of
how many
participants
GPU
for research
resources your
studies which
models
are
are using.
involving socially
With one
as
line
well
of
as
code,
racial
you
and
can
ethnically
visualize
diverse
model
groups.
predictions in form of
I
images,
would also like to lay
videos,
emphasis on the
audio,
fact that this is a
kilometer
highly
charts,
advantageous
molecular data,
time-efficient approach
segmentation
in the
maps,
sense
and
that studies
3D
which do
point
not
clouds.
engage in social media
You can
also
save
require higher
everything you need
motivation. That is
to
more phone
reproduce your
calls need to
models
be placed,
days,
more
weeks,
in-person
or even
appointments,
months after
and
training.
longer wait times than
Finally,
entries
with
which can be
reports,
made
you
just
can
by the
make your
click of
models
a
come
mouse.
alive. Reports are like
So, for
blog
instance,
posts
a
in which
survey-based
your
research
readers
study which was
can
conducted
interact
in
with your model
2018
metrics and
concluded that
predictions. Reports
the
serve
incidence
as a
rate
centralized
ratio
repository
for the time
of
to survey
metrics,
completion was on an
predictions,
average
hyperparameter stride,
3.31
and
times faster
accompanying
in
nodes.
social media
All of this
participants as
together
compared to
gives you
intercept
a
participants.
bird's eye view of your machine
Social
learning
media
workflow.
recruitment strategies can
You can
also
use
target
reports to
ads for
share your
display to
model
participants
insights,
of the
keep
desired
your team on the
population
same
head on
page, and
and
collaborate
allow these users
effectively
to respond to the
remotely.
online
I'll leave a
request
link in the
surveys
show notes below
immediately
to
and
help you
without
get
the
started.
requirement of
And
in-person
now,
contact
let's
with the
get back
research
to the
team,
episode.
thereby
And
advocating
you've worked
for
at
a lesser
some big
stigmatizing,
companies too. I
a
guess
lesser
like generally like what
anxiety-provoking approach,
kinds of
as well
problems
as a
do you
more
see
cost-effective
Pranav,
and
when
your
companies
time
try
is
to
over.
take research and productionize it? Like what are the main
Okay.
ways that you see companies fail at this?
I think one
Okay.
of the
So
big
now
things
the
is
opposition team
a lot
can
of
begin with the
companies
opening
are
statement.
chasing buzzwords.
Hello, I'm
For example,
Upasna
Chasing
from Team
buzzwords?
9.
Yes.
Before I come to my
I
So
see.
own
everyone's like,
arguments
yeah,
today, let me
how can
first
we use
dissect
BERT? How can we
what the motion
transform
actually
it? And
means.
And you can
Although
look at them
social
as like,
media might
you
play
actually
some
don't
role,
need
which
that. You
the
don't need
proposition
deep
will
deep
undoubtedly
learning. Like a lot of the
compel you to
problems can
believe,
be solved by
but to say it is
traditional
crucial is
classical
an outright
algorithms.
and blatant inflation
So
of
sometimes
facts.
companies
Something else that needs to be addressed is the aspect
yeah,
of
just want you to
engagement.
use
There are two
very
ways in
fancy
which we engage with
technique.
scientific research.
It,
The first
the
aspect
reason
being
can
the
be because they
general
don't really
study of
understand
the scientific
what is
literature and the
happening,
culture
because I think there are
as
a lot of
consumers, and the other being the
there's a lot
individual
of
engagement of
misunderstanding
respondents
in
and
AI
researchers
reporting.
with their subject
You
material.
know, like if you
A
see like a lot of
meta-analysis of
journalists
over
or reporters
1,100
talking about
studies and
AI,
about
and if they
9,000
don't have
surveys
background
by Meng
in
Jianwu
AI, then they
found that
might
although the number of online
simplify
surveys in
or just
published
doesn't
literature
exactly
grew
like reflect
steadily in
what
research years,
exactly is
even
going on. And the
substituting
second is
other
some
types of
companies
surveys,
might just
the
want to use
online survey
buzzwords to attract
response rate
clients. You know,
remained
like
at
it's like, oh, we're using
44.1%.
state of the art
In the spirit of
techniques.
scientific research,
Then
we
yeah,
as a team
so
conducted
some
our
people,
own
some
survey,
companies actually
and the
like
results
go out their way
replicated the
to
meta-analysis
try to
and
use that.
unequivocally so.
So that's one
With our
problem. I think the
responses numbered
second
at
is
87
that
of the 1,200
so
participants that we
probably
sent
was
it
a
to.
lack of data, and I think Lucas
Sending an
explained
online
that
survey
very
to
well,
more
because
participants
you
did
work
not
with
generate a
you
higher
also try to solve
response
the
rate,
problem,
a
right?
potential argument against
So
the
in
supposed
research,
diversity that social media
people work
offers.
with very
In
clean,
fact, a
static
clearly
data set,
defined and
and
refined
you need
population
data set to be clean and
with the
static
use
because
of
you want
other
to
types
focus on
of
developing
surveys
model.
or
But
substituting
in
social
productions,
media or
you would
online
have to
surveys,
usually
we
have
led a
like
higher
I
response
think
rate.
as models
Coming to
will
the
be more
other
and more
aspect of engagement,
commoditized,
as
and you can
consumers
speak
of
off the shell
research,
model.
we've
So
seen
the
that
bottleneck
numerous
now is
challenges and
data,
controversies to have recently
and
arose.
real
A
world
study
data
by
is
Boston
nowhere close
University
to
and
like
another by the Journal
that
of
like
Health
research
Policy
data.
Research found
So the problem is
that
like
the more
collect data
frequently
and
respondents
verify
report
data,
posting and sharing any
how to
info
cope
to their
with
social media,
constantly
they were increasingly likely to be highly
shifting
misinformed with
data
respect
drift.
to the subject material.
So there's
The
a
ones
problem
that
with
come to
data.
mind
And
are
another
the
problem is with
antimicrobial resistance crisis or the polio vaccination epidemic in Israel.
So in
While
research,
social media
like
might
sometimes you
act
don't
as a tool
really
for
care more about
adjuncting
like
research methodology,
state of
it
the
is
art,
definitely
little
not
boat.
crucial. That is a
Basna, your
hyperbole if
time
I've
is
ever
But
heard
also in
one.
real world, like you
Thank you,
sometimes
Anna.
like if you would be like you just
Okay,
don't care
thank you.
about
So we'll move on
the
to the
kerosene
rebuttal round now.
or F1 or whatever metric
The
you pursuing, but
the
just care
second
about like
speaker
how
of the
can we
opposition team can
explain
start
the
by asking their
decisions
questions. You have
that
1
the
minute
model
to
is
ask your
making.
questions. You can begin.
But I think like a lot of them like
Before I
people
start, I
are,
had a question. Do I have to ask all my questions
I think
in
a
one
lot of
go,
the
or
people are like
will I
focusing
be
a lot of
waiting for
time
them to
on.
You have to ask all your
So I
questions
think we are making
at
progress.
once? Okay, so the first
But
question
I
that
saw a
arises
tweet
in my mind
recently,
for the opposition is when you speak of
I
cost
think it was
effectiveness
someone at
in terms
OpenAI who
of
was
social media
arguing
being
that,
less costly.
that folks
In the studies that
should
in the
not
survey
teach
that we
anything
conducted as
besides
well as the
neural
study
nets
Oh my
done by
god, it's such a
Meng Jianwu,
clickbait.
it was
And
shown that
it's funny because
respondents
it
are
reminded
most
me of
likely
when
to
I was
respond,
in
overwhelmingly so, over 70%
when I got my
are more
first
likely to
job out of
respond when you
school.
give them a
There's
monetary
sort of a similar
incentive to
debate,
respond.
but it was like different
Secondly, when you
topics. So
mentioned
like
that
it's
with
basically
social
like
media you can
machine
use
learning versus
advertisement to
rule-based
target
systems.
certain
And there were a lot
subsets
of
of
kind of older
populations.
researchers
You also have to consider that
who had kind of
advertisement on
bet their
social
careers
media
on like
also comes
logic
at a
and
cost.
rule-based
And
systems. And
what with the current
and
rates
they
of
would say,
Instagram
oh, like,
and
you
Facebook
know, it's like
advertising being through the
obviously
roof,
like you should do both.
I
And
don't
I was
think
like,
it's very
come
feasible
on,
for
these
any
rule-based
researcher
systems don't
to
really
go down
work
this
in
path.
anything. And
Secondly,
like,
when you
can
talk
you
about
find
an
me like a
ethnically diverse
benchmark where
group
it actually makes
of
sense
people,
to do a
what
rule-based
about
system?
the
And
ethnicities that are
I was thinking
not
that's
traditionally
how I
known for
felt at the
having
time, and I
access to
think I
internet
might
or
still.
not being
I mean,
traditionally
you don't see a
technically
lot of
literate?
rule-based systems.
For example,
in,
how would you do a
production in
survey
the
about the
last,
quality of
decade
life
or
in
two.
a tribal Adivasi population through social
Or
media?
I don't come across
I don't think
them, I
any of
guess.
them have access to
And
internet
then I
or
was
social
thinking,
media for that matter.
you know, when
And,
that person made that clickbaity tweet, I was
Your
like,
time
no,
is
no,
over.
that's ridiculous. But I was wondering, am I now like the old guy who's like trying to just
Okay, so with
justify things
regards
that I know about?
to okay, so can I
No, did
answer?
you
Yes, you
did you
can
get
start.
baited? Do you participate in the discussion?
No, I'm like always
I'm
afraid of
sorry, can
controversial
you
topics on
once
Twitter.
again can you repeat
I
the
just I
question?
never.
Sorry,
Not even controversial. It's just wrong. I mean, like I don't get it. I mean, if it's not from some OpenAI,
can you repeat
I
it?
would think that
I'm sorry,
person was
Anuj, we cannot do
trolling. I
that.
don't know, but I don't know. Maybe he's trolling.
The
I'm
1
not
minute
Okay.
time
sure
allotted
anymore.
for the
But it's
question
funny
is
because
Okay,
not
I think your
so
work is
yeah,
mostly in.
so with
Unlike
regards
my
to
work,
the
it's,
finances involved,
if you look
there
at my
was
own
a study
research, it's not
conducted
in neural
in
nets
which
at all.
LGBTQ
I mean, you actually work in neural nets, so I guess
people
what are the
were recruited.
non-deep learning algorithms
So
that
in
you
that
think are useful?
already
keep around?
around
Why
1,300
why would you use
people
a
were
different one?
recruited using Facebook and Instagram ads.
I mean, like,
whereas
like
personal
actually Boost
intersection
is still like
recruited
the most
around
popular
say
part of the
2,300 people.
tackle like
and it was found
competitions.
that
So a
social
lot of
media
them are still
recruitment
very
accounted for
good,
only
like
2.2% of the budget,
neighbors are still very
whereas
good for
the recruitment
anomaly
costs using
detection,
personal interception were
so a lot of like
97.8%.
really great algorithms.
Now,
Like, a lot of
this
people
is a
nowadays
massive
don't even
difference,
know
and
what
social
boosts
media was
look
also
like
3.1 times faster.
so which is
Now,
really
in
sad.
this present day scenario, when there is like
So yeah, so a lot
20%
of
of
things.
studies are called off because of delayed
I think
recruitment and
it's
22%
the other day my
are called off
friend was
because of
telling me about how I
financial
was interviewing
constraints,
somebody and
wouldn't
that
this
person could explain
factor
perfectly well
of cost
what the
effectiveness
transform
be crucial in the favor
what
of
a
social
transform
media?
model is, but can't explain what a decision tree is, and it was just a bit, I don't know. Maybe I'm
Okay, if
old
you're
too.
done and your
I
other
don't even
teammate
know
wants to
anything.
add something, they
I
can do it.
would,
Or we
yeah.
What are the situations where like you would
I
recommend
would
using
also
like a
like to
boosted
add
tree
that you
versus
said
like
that
a neural network
internet
approach?
could be a
I
barrier
think
in
definitely for
conducting
base life, for
social
example, right?
media-based recruitment studies.
If a
But
simple
I would
model
like to
does a
add
job
that,
reasonably
recently a
well,
study was
I
conducted, a
mean, this
systematic review,
always
which
values
was
in
conducted in
trying, but like in
2018 by
production,
researchers in the United States
the
of America.
simpler the
They
model
concluded
is,
that
actually, it's
even
easier
homeless people had
to understand and to
access to
implement
social media accounts
and to
once a
avoid
week.
mistake.
80% of homeless
I see.
people
So
also
like
had
if you don't
access
get
to internet
improvement
about
from
at least once a
like
week.
deep, like more
And
complicated methods,
considering the fact
don't go
that,
there.
as I said, Facebook has nearly
I mean,
2.94
like,
billion
it's also
active
hard to
users,
tell because
there is a
a lot
vast
of improvements
demographic
great. So you can
as
say
well
that,
as
oh, this only give
contextual
like
diversity in the sense
1%
that
improvement is
it
not
is
worth it. But even like,
extremely
for example,
easy to
1%
find
improvement,
out
if more time investing, it can
participants
be
of
like
the desired population
another
for
and
your
another
study
and another,
using
and over
social
time you can
media
get up
as
to 10%
compared to
improvement. But then
traditional
if you like
studies
just
which
like
do not
stifle it from the
engage in
beginning,
social
then
media
you
at
will
all.
never be able to reach the point where it should be.
Okay, so
So
I
I'm just
would
saying
also
this
like to
like
add that Facebook and other social media
I'm not pro
platforms,
like
especially
not using
Facebook, has a
deep learning. I'm actually
basically
very pro
has
deep
a
learning. I'm just
advertising
saying
program
that
that
to start
offers the
we should
ability
not
to target
forget like
advertising
simple
campaigns using zip
baselines, and
codes.
I
So
don't
for
think we spend enough
the
time talking
studies
about
could be designed
baselines
to
or
target
implementing baseline.
ethnically diverse zip codes
Interesting. A
throughout
lot
a
of people have
particular
said that.
location
I
in
happen to
order
agree
to
with it.
increase
But
the number of
if someone was going to
participants from
ask
particular
you,
ethnicity.
why are baselines important? How would
Also,
you
studies
answer
had
that?
been conducted on pregnant
Because
women
like a
who
metric
are
by
less
itself
likely to be
doesn't
seen
say
in their
90%
early stages,
accuracy
especially,
doesn't
and
mean
these people
anything.
could be reached
So we say,
using
oh, my
social
model
media.
is like
And
is amazing. Can
there are
like
other cases
it
also,
has like
such
this
as
accuracy.
stigmatized
It's
cases.
like what does it
For
even mean?
example,
And
lung
for
cancer
example, like
patients who are
somebody
chronic
showed me
smokers,
this
they
model is like
prefer
90% accuracy, and
communication
say
via
like,
social media
well, if
because
it's just like
it
make it
reveals less
random,
of their
it's
personal
like
details, and
it's like
they also feel
eighty
that
9% accuracy
it would
already. So
lead to
what's
lesser
the hope? What is
stigmatization
the
because
point of like getting this?
331
So baselines
smokers,
are
people
I can
of
say
this
baselines are landmarks to
whatever
help
these
you
participants, 331
localize
of
and
them were
where the
recruited
model
using social media
performance
compared to
and
only
where you want to
30
get to.
using traditional
So something
newspaper
very
ads.
interesting, like human baseline, for example,
Yeah,
like
I
what
guess
like
so.
if human
Underrepresented
do on social media,
minorities
like
also
what
have
Your
been.
time is
they
over.
could do. So it's like maybe it's like 90% is
Now
also like
the
really amazing. If the human
proposition team
baseline
can
is like
start with their
55%, so we say, oh,
questions.
this is like superhuman performance. But even so, if the human base like 99%,
Okay, so again, I'd ask you the same question with 22% of sorry,
So it's
with
like the
22%
human
of
baseline is kind of like
studies
maybe
being
in some
called off due
sense
to
like
financial constraints, and a further
best case
20%
scenario.
being called off due to
I
delayed
think
recruitment.
in some cases, in a lot
And
of
social
cases,
media
the
offers,
humans
as per a
baseline is
study,
oracle, but it's not
3.31 times
always.
faster recruitment. Wouldn't social
Do
media
you
be
want to talk?
crucial?
Another
Also,
thing I'm kind of
with
curious about
regards
is
to traditional forms of
your work on. I mean, just because I think
recording in
like,
progress.
Fake
how
news
would
is probably
you
going to be a big topic
target
again with the
specific
election coming
rare
up.
cases? How would you
I
find
think
them?
this is a
It would be
fun
much easier to do so on social
class
media.
project.
Also,
So it was after the
social media has
election,
played a massive role
and we're
in
curious to see. So
propagation of
you know, like I
research,
think echo chamber can
dissemination of
also help,
informations, increased citations
like
of articles,
echoing like fake news,
and
right? And so like
also
the same
increase
fake news usually circulating the
site
same
visits and
echo chamber.
increase what can I say, full text article downloads. Would you still
And you know,
say
like
that it's not
if
crucial
one echo
for research
chamber
engagement?
shares a certain piece of news, and another
The
echo
opposition
chamber
team can
shares
start
similar
answering
news
the
but with different perspectives, there might be some. I'm not sure how helpful it's
I
going to
think
be,
I'd
but
like to
it might
start
be
off by
interesting to
answering your
like
last question
cross-share.
first. So
Like
it has led
brings
to an increase
the
in
piece of
downloading
news
full
from
text
one's
citations.
perspective to
So
another
I
echo
think
chamber.
you are
Not fake news,
confusing
of course. There's no point in
social media
spreading
with
fake news
the
from
internet.
one
The social
echo
media and
chamber to
internet are two
another.
separate entities.
So
And
what
unless
we
you believe
did was that
PubMed is a social media platform,
we got
like
a
I
lot of
can't.
tweets and from hashtag.
I don't know, but
So So as a time was
internet,
after the
while social
elections,
media
we
is a subset
collect
of
a
the
lot of
internet, it is
tips
not the internet
from
itself.
during
The internet has
elections in
increased
one
access to
state.
these resources starting from
And
the
what we
90s, ever
did was
since
we
the
have some
internet has been
seed
around. And I
hashtags
think
that we know
another one,
was
you
like
raised
whether
a
it's
point where in chronics, so you said
pro-Republicans or
about targeting
pro-democratic.
rare groups and people who are not
So from those seed
able to
hashtags,
or
we
who
have we
feel
make assumption
safer
that
responding online.
if
There was
two
actually a study
hashtags
conducted by
belong
Boynton,
in the
which
same
found
tweet,
that if you
then
relate
they
it with
are
the
likely
meta-analysis
with the
conducted
same
by
sentiment, right? Like
that we just talked about, they actually found that of the 60% of respondents
like
who
if
do
one
not choose
hashtag
to share
appears
their
next to an
information
hashtag
via
that's
social media do so because of the fear of their personal information being shared online.
So from that,
and I think we all know
had
how
an algorithm
important
which
it is,
like,
how important your social media
just
presence
to
is today.
resolve
So I
conflicts.
feel your
It's very
personal
simple, like
information
majority
being linked to the
voting. So,
internet, no
and
matter
so
how
we
much
will
safety you
able
as a researcher can guarantee,
able
is
to
going to be
like
detrimental. And I
label
think
about like
Sarthak can take
1,000
your last
hashtags.
question.
And so from this, the number
not
of
trusting the
conservative or
computer.
liberals is
And
not
when you
just not
compare
that
that to
high.
something like lung
But then
cancer
about
and you
25%
call
of
them
them are like
a
conservative.
significant
For example,
minority,
like the
my question
number
to you
of
is which one is the greater minority here?
members
Do you
are
think
like
there
more
are
than 3 times higher than like
lung cancer patients, of course,
Democrat,
being the
and
minority here?
25%
The question
Democrat,
is, which subgroup do you think is less representative?
So you found
is
the
it
echo
the teenagers?
chambers.
Is it the young adults who are facing
So
sexual
we
health
found this
problems
like
who want to
I'm not
consult
sure
their
it's
physicians?
echo
Or is
chamber,
it the lung
but
cancer
but
patients?
it's definitely like people who share
And,
similar beliefs.
yeah, that
Definitely
is all
have
I have to
stronger
say for now.
ties and
Thank you.
people with like with
Now
with
we'll be
different
moving
views.
on. So I would like to hand it over to the judges for their questions. We
So
can
then
start
were
with the
you
questions for
suggesting
the
to sort of like
proposition
spread
team and then
information
move on to the opposition.
between these communities, or
So we never
Should I
had
start
access to that because it would require live.
You
don't have to ask.
Yeah, we would requiring having access to certain social networks. But we would idea. So we ran
I have a
into
question
some
for the team which was propagating the use of
literature,
social media for
and
research.
they say this like
So there was a
actually
lot of
like if
talk
we
about
show somebody
use of
the
social media for
opposite, like
promotion and
a news
advertising.
article with the
What
opposite
I would
point
like
of view is
you
not actually
to tell me
not.
is that
If
how reliable is the data generated from this
going
use
to
of social
ignore it.
media?
So
And
if
do
you
you
believe
think
in
the
A
use
and
of
I
social
show you
media
the
for
opposite of
research
A, you
kind
was
of
like, oh,
dilutes
that's
the
fake news, right?
sanctity of the
So the kids
research
you
being
can't you have
conducted?
to like slowly
I
show
would
them
like
it's
to
like
know your views on
similar
that.
but like slightly different. Like, you can't
Thank you for
you
your question,
can't
ma'am.
give people a totally opposite viewpoint and
Sorry,
expect
someone
people
was speaking.
to listen to it.
I see. That makes sense.
Thank you for your question.
So we never had a chance to like test out the algorithm. But we hope that like
yes, to a
but we
certain
thought that
extent,
like
survey-based
detecting
studies
echo
may come a
chambers
little
might be the
down in
first
the
step
evidence-based
in
pyramid of
finding a way
research,
to
but it is
break
also
them.
necessary to
Uh-huh.
understand
I
that
see.
low
Yeah, yeah.
quality and duplicate responses can be
Although maybe
efficiently
people
removed
just
from
want to
the
live in their echo
data
chamber. I
set,
don't know.
in the sense that low quality
I think
responses
Silicon Valley
have
is a
been filtered
massive
in the
echo
research
chamber,
studies that
really.
we
For
looked
sure.
I
up
think we
on
all live
PubMed
in the
by,
bubble.
like, IP addresses which were
And I feel like
associated with
somehow like
malicious
this
accounts
pandemic
were
also makes me
removed and were
realize
not
like
considered
to
for
show like
online survey-based studies, and
how
even
different
duplicate
our bubble
responses
is,
in the
like how
sense
strong
that
it is.
multiple
Okay, another
responses
just
which were
question
put
I wanted to
forward
ask you
by
about. So
the same
you've
IP address
you've
within a short
recently
window
gone
of time
from
were also
a
I'm so
removed
sorry.
from the
like a bigger
My teammate
company
was kicked
to
out. I
a
think she has
startup.
just texted me. Can you please add her back
Which is
in?
a little bit of a shift. Has there been any
So sorry to
surprises there?
interrupt.
Has that been like I guess like how does that
She's
feel
there in
to
the
go
meeting.
from like big
Yeah,
company
I
to startup?
think
Is it a
I'm
big like
very sorry
cultural
again. I think there was
shift
a
or
technical glitch and I was removed.
It's a
same
big
bubble?
different.
Okay.
It's
Sorry to interrupt,
such a
Anand.
big
You can
different.
continue.
no problem,
I
ma'am.
think
So
and that's what I
what I
wanted,
wish to
right?
say
I
is
thought
that
that
these studies that we
after
consider should
graduation, I
not
would
be
like to
taken,
try with different working
should not be
environment to
considered
see
in the
which one I
research
would
are
like.
actually filtered
So
out
living NVIDIA
by
was not
various
a
mechanisms
reflection
in
on
the sense
NVIDIA. I
that,
just like
as
to
I mentioned,
reflection on
malicious accounts as
myself
well
because
as
I just wanted to
multiple
change.
responses are
Totally.
easily filtered out by a
Yeah, and
number
my
of
coworkers
softwares and
NVIDIA have
services.
been extremely helpful, and yes, they are really kind.
Oh, ma'am, also, in case of
So
larger recruitment, as you said,
it was a bit shock
sorry,
to join a
there may be
startup. I think it's
unwanted
definitely the
people over
first
there.
is
So,
it's a
ma'am, there
workload.
is
It's so much more.
It is
So
so
you know
much
how-
more
survey also
interesting.
has a robust
which is a good
inclusion and
thing.
exclusion criteria. So all
So
those
you
who
know,
do not
at a
fit
big
into the
company, you might
study
see people like
parameters
leaving
would
home at like leaving
themselves
work at
be
like
excluded. While
5,
social media would
6.
play the role of
And at startups, you might have
attracting
people like
participants,
you
it would
might
not
you might
play
got
the
like
role of
PR requests
screening
at like
them through.
midnight on Saturday, right? Which
Thank you for your response.
Which is not a bad thing. I don't know. I think
which was speaking against the motion.
I'm still very ambivalent about on the work-life balance.
Am I audible?
Discussions, you know.
Yes.
Some people say like, oh, you
So
shouldn't
basically,
like
social
work
media,
on
I
the
think you would agree
weekend,
with me that
or
okay,
like
doesn't
no
help with
company
literature
should
search,
expect
like
the
you pointed out,
employees
but it
to
does get you in
to
touch
work
with
on a
subject
Saturday
experts
evening.
and get
But
you a
the
lot of
point is like
constructive
companies
feedback
might not
on your own
expect you.
ideas and help in generation
But
of new
it's like
ideas
whether
as
you,
well.
what you expect
Okay,
out
in
of
terms
yourself.
of the
And I don't
volume,
want
velocity, and
I
the
don't want
variety,
you
what else do you suggest as a viable alternative?
I don't want to promote the culture of like you have to work hard, but I do believe that when you're early in the career, there are certain compromises you might have to make.
Do you want to
which depends on like,
sorry?
what you want out of
Do you want me to
life.
repeat the question?
No, man, it's
Anyways,
fine.
that's a very
So
roundabout way of
social
saying
media,
that
as you
I
said,
think
we are
can.
using it for constructive feedback. We are using it to get in touch with our peers and to
It
have
sounds
them
like you're
take a
kind of
look
enjoying
at
that
what
experience.
study we're doing, how we're processing the data.
I also don't have
But
much alive,
the
you know.
crucial thing I would like to point out here is not the social media aspect of
I
itself
guess right
in
now
research
there's
engagement,
less.
But what
less
we are
going
talking
on,
about
for
is
sure.
constructive criticism from our
So
peers
yes,
as
the
well.
big shock is that
This
like
can be
yes,
achieved without social
people
media
work on
quite
the
easily.
weekend,
Even if you
and,
were to, let's say, give a phone call to a
and I
group of
quite like
colleagues,
it.
or
I'm not
as
sure
a
how
student,
much
I would
longer
take
I
my
would
paper
like
or
it
take
because
my
I heard
project
that like
ideas
as soon as you like have
to my
family,
department,
like for
to
you
my
guys,
ethics
like, I
committee,
heard you had a baby
to some of the senior
recently,
researchers in my
I did,
college,
yeah,
and then
yeah.
they would
Do you
help me
still
out,
go
they
on the
would guide me
weekend?
better. And I think that
You know,
on
I
social
guess
media,
like
if I were to make a post, let's say on Facebook,
I
I just
don't think
post
I
a
have like
link
a
about my
very
study and I
strong
have one of
point
my
of view.
friends comment on it.
Like you know,
If
I
it's
feel like
open,
it's a
there
little
might be a
bit
lot of
weird to
there
tell
might be a
people
lot
not
of
to
white noise,
work
there might be
super
a lot
hard.
of
Like I
background
worked
noise, some
incredibly
comments that are
hard in my
not
20s,
so
and
not savory, to
I
say
kind
the
of
least.
like
Some
to
comments that are
imagine that
not
hard work
exactly
pays
helpful
off.
to me in any manner.
I
Whereas
think
with direct person-to-person contact,
I
I can
proud of
always
the stuff
ensure that
that I
I'm
did.
going to get an
I think,
objectively
like,
good and constructive
You
criticism
did, like,
or
you did a
constructive
great job. I mean, I
response
have done a
from
lot
this
of great
person, and it's
things.
going to be
Oh, thank
a
you.
a
I mean,
fruitful
I
interaction.
don't know. Like
I'm
I
a
think
fan.
I
And
don't.
with this, I
If
would
you're
also
lucky
like
enough
to
to
add
have
a
a
statement
job
to this
where,
answer to bear in mind our
I
stance
think
on
there
this
is
topic as a team. Is that
in the
social
best
media is not
situation, working
crucial
really
for
hard can
research
be like
engagement. It
incredibly
does play a
fun.
part
Like
somewhere.
for
We
me,
must agree
so
that it's,
often.
let's call a
And I
spade a
realize,
spade.
like,
Social
I
media does
I
play
run a
some
company, and
into a part in some extent
like maybe
in social
I'm
in
coming from
research
a particular
engagement,
point of
but it
view,
is
but for
definitely
me,
not
like
crucial.
working
Thank you.
really hard, it can be like a real joy. Like
If it's okay,
when I
I'd
actually
like to
started
add to what my
my second
teammate just said.
company,
I believe
one of the
social
really fun
media
things for me was that it actually made
for
sense for me to
the purpose
like,
of collaboration
like,
does offer
pull
a
an all-nighter
platform.
once in a while.
But what
I
it
hadn't-
also
I hadn't
takes
done that
away
in a
Really?
is
Do you
the
still
kind
pull
of
all-nighter?
subjectivity that
No,
you
I have a baby.
have
It's
in
like,
our
well, I pull a different kind
This
of
is
all-nighter.
natural, natural all-nighter.
Yeah, natural
to
all-nighter. It's
send
an
your
unproductive
own surveys
but
to
I also
a
think
very
like
selective group of people,
think
or
that,
someone you just found
you know,
off of
the-
social media, and you
I mean,
were
the
to
important
forward
thing
a
is
link.
like a company's
There's
trying to
actually a
do
study
something and
conducted
like
by the
you know
University of
figuring out how to do
Pennsylvania,
it
which looked
is the important
into
thing, and like figuring out how to do that over
surveys
the
being
long
sent
term
to
is so like
research
it needs
professors
to
in
be a
various
sustainable pace
universities across
because
the US.
like
They actually found
you know,
that they were,
if you
for
work hard
about
and burn
80%
out,
of the cases, they were
that's super
anyway not likely to
counterproductive. But I
respond
don't always
to
think that
links like
burnout
these.
actually
So I
comes
believe
from
it doesn't really
like working hard. I think burnout
add
comes more from like working on things that
as
seem
much
pointless or
of
like
a benefit
you're
Yeah.
not seeing
as
the
we would
success that
think
you
it would.
wanted.
But of
I
course,
see.
like my teammate said,
Yeah,
as
that
simple
makes
as that,
sense.
it's not crucial. You
Yeah.
do have alternatives
Also,
to
I think
reaching out to
for me,
people
like seeing people
without the
working
use
on
of
the weekend
social
is not
media.
a bad thing. It's just like
And
even
although it
me, like
can
I
be
am so
beneficial,
feel
it can
motivated to go on
also,
the
on the
weekends
other
just
end,
because
be
I
detrimental
realize what I
if you
do
do
and
not
I have
reach,
faith
if
in
it
it.
does not
And
reach the right group
I
of
think
people.
I also want to contribute. Like if on a Saturday night I could stay at home and watch some like I don't know.
Thank you.
Some I was a bachelor. I don't know watch it, but it's
Okay.
just what people seem to be
Anand,
watching.
question is to the
Or I could like just
team, I think,
go on
proposition team.
our GitHub and
See,
check out some
largely,
PRs.
your argument was that it can
I
be
know
used
that
for
sounds
research.
like a
Both of your
horrible
teams focus mainly on surveys.
analogy.
And
But yeah,
using
so I think like yeah, I
social
feel
media as a
motivated. So it's
sort of
like okay. So anyway,
recruitment
back to the question. So the
And there is
first
no doubt
thing I
that
noticed is
results in serious
the
people
different
selection
workload.
bias.
And
People who respond
yeah,
are
and I
very-
feel
are
and
really
I
not
feel okay working on
representative
the
of the larger.
weekends because I'm still like
So
I can
my
still talk to
question
my
to
coworkers on
one
the weekend
is
and I like it.
team to
And
Well,
it's also very
survey is
understanding
just
because
one
like
form of
because people.
many other
So I
research
think
designs.
everyone knows that other people,
I would like
some
to
people
hear
work on the
from you,
weekend. Some
how
people
can
also
social
like
media be
give some
used
more
in other
flexibility.
research
Like
fields?
if you even on the
Beyond
weekday,
survey.
if you feel like burnt
That's my
out
question
and like
to you.
tired and want to take a day or two off during the week, and it's fine as well.
Also, there
So I
have
think it lies
been
understanding
previous
like the
studies
making the
where
schedule to work for you.
the
Don't
social
necessarily
media has
follow
been used as an intervention where
a typical
it has
like
been
5
used
day
in
work
controlling
week and
smoking
like
per
work on
se.
the weekdays and then take weekend off,
Yeah,
weekend
that's
off.
what I
I think the
remember.
second thing
And
is
other
the
than
amount
that,
exposure I have choose the
that's
entire
all
stack.
I know at this point.
So working in a big company, I'm most focused on very specific product, and I don't.
So I
I'm
would like to
shielded
add that
away
our
from it one other
opposition
aspect,
team
as in like
actually considered, sending out their own
yeah,
survey
as in like QA
social
or
media
like client.
recruitment for research
But at startup,
engagement.
I get a chance to
But
see everything, and we are sort of
to
building
say
this like
that
from scratch.
research
So I'm
is,
so like
social
exposed to the
media
decisions
like
that we have to
Facebook,
make. For example,
Twitter,
like
Instagram,
what
they are
what's the ICD
absolutely
tool to
vital for
use, or
research
like how
engagement.
we should
In the
structure a
sense
repo.
that
Like, do you want like a
I
mono-repo,
could
or do
actually
you
give a
want
live
like
instance of say
various
Norm
repos?
Labs, which is an Instagram
So
account,
and also like
which
these like decisions on like
recruits
what
these
tools you use, because
patients
when you join a
for
big company, usually you
psychiatric
have to use
studies,
standard tools,
and
and
it
somebody
has
already
over
made the decision
10,000
for you. But
followers
as
on
a startup,
Instagram.
you also
So
have a say
it's not only
in
about
choosing the
surveys,
tools,
it's about
which
reaching
expose
out
you
to
like
people,
various
and
problems as well.
if
So I really
they
like it.
if they satisfy
Yeah, and also
your
of course it's
inclusion
like
criteria,
the size.
then
Like at
you
a big
could recruit
company,
them not only for survey-based
there are a lot
studies,
of
but
people.
for
I think it's
other
nice at a
larger
big company
studies
as
on
in
the
you have
evidence-based
access to a
pyramid.
lot of people. But at a startup, you have
Okay,
a
thank
small
you.
number of college, so you can't
My question to the
randomly
other side
message
is
somebody
the
from another team and ask for a
see,
coffee, because
and this focuses
only
more
have
on
like these
dissemination,
people
using
on the
social
team.
media for dissemination of research
Cool, makes sense.
results.
And
largely,
all right, so we always,
I
so I think
think
we're
made a
kind of
comment that
going over time, and we
more
always end with two
use
questions. I'm really
information,
kind of
use
curious to hear
information, those kind of-
your
rather
takes on
than
these.
pure
So,
science based.
yeah, the first
So my
question is that
this thing is that
what
that
is
actually
a
reflects a
topic
limitation of
in
the scientists. They have not
machine
learned
learning
how
that
to
you think
use
people
social media.
don't talk
They have to learn to use
enough
social
about, or
media
they don't
better.
talk?
So
It's
it's a
an
problem with
underrated
the
topic that
scientists
people
rather than
should
with
talk
the
about more, but they
platform.
don't.
That's my argument.
So
and
I think I have
you
a
can argue
list of
otherwise.
things I usually like throw around. One
right.
of them usually one of them is
Awesome.
a graph. So I love graphs.
Sir, I think
used to
that
think of
social media
graphs
is
so
a
underrated.
potent
I
tool
think I had a
indeed.
tweet
And
about, like,
what
3
you
years
said
ago
about the
about it.
scientists being unable to use social media,
But I think it has changed.
well, yes,
like,
that
I
is a
was at
problem that
Europe
needs to be
in
addressed.
December, and I saw
And
so
definitely,
many
if the
papers on
person
graphs,
is
and
inept
there's
at
a
using
workshop
social
on
media
graphs.
themselves, then the data that
That was
they
like the
gather
most
from it and
technical
extrapolate for
shock.
the
So
general
I
You're talking
population
about the
think,
is
computer
also going to be
science sense
tainted.
of graph.
But that just goes to show that
Right.
in today's world,
Or do you mean
graph,
like literally
as
also
the
like
world
graph
exists today,
network. So
Like a
the
graph
way that social
theory.
media works
Yeah.
is that you cannot
So now we have like
truly
they have
ever
like
have
several
someone
like
fully
there are a
understand
lot
it.
of
The
graphs
complex
like
algorithms,
GNN or GCNN and
the
graph
insider
convolutional
information,
network.
whatever happens
So I
behind
think
our
like there are
screens is
multiple
truly
uses
too
of
complex
graph
for
in
someone, let's say, of our
deep
medical
learning.
specialty
First
to
is
sit and
as
specialize
so
in
graph
again.
is natural
And one small statistic
representations
that
of
I
like
would like
many
to bring up
inputs,
for you,
right? So you
sir,
have a
since you
data
mentioned
administration
that
network,
both of
for
us
example,
teams
is a
have been
graph.
focusing a little
Or
on
recommendation
survey-based
systems, and
studies.
we have like
This
users
is
and
from a
items, and it's a
study by
bipartite
Dr.
graph.
Chittaranjan
So
Andrade
graph is
himself, who is a
natural
prominent researcher
representation of
at NIMHANS.
and
And
also
this was
like
published
graphs
in
can
the
also like
Indian
output.
Journal of
So it's not
Psychological
like
Medicine.
any so a
He
lot of
said that
distributions can be
since 2016,
represented using
where
graph, like use pictorial graph.
online survey
So graphs can
returned
be like can represent both input and
1,682
outputs.
results,
And also like
the number
graphs
has far
can also
grown
have
and
a
it's
lot of
become closer
relationship
to
with
4,400 in
convolutions. So they're
2020
both
alone.
local.
And with the rise of the pandemic, I think this number might have
So
gone way upwards
yeah,
since
so they
then. There
both
has
focus
been
on the
a
local
stagnation
connections.
in the rate of growth for
So like
meta-analysis,
graph, like
a slight decrease in fact.
once it's
And in
neighboring
randomized
points, and
controlled
like
trials, there has
convolutions
been
when
a
you have
decrease
this
by a
very
large
local
margin. Therefore, we are moving
linear
towards a world
transformation.
where this contactless survey-based
I'm
study
very
pattern is sort of
hand-wavy
becoming our
explanation right
primary
now.
form of research.
So yeah,
And
so
in a
I
time like
really
this,
love graphs, and
the
I'm
results
so
that
happy
we
to see
can
that it's catching
gather
on.
from the current literature that exists is that definitely social
So I
media
think
does
like
not play
other things
a
underrated
crucial
is like
part
engineering
in
aspect
research
for
engagement.
machine learning. So I haven't seen much for talking about like integrations
To add to what Sarthak said,
deep
actually,
learning,
I believe
or like
no matter how
version control.
adept
But I think
a
all of
person
those like
is to
people are
share
so like
their
I'm
research,
not
they're
unique. I
at
feel like
the other end of the
what I
spectrum,
see other
the
world I've
purpose of
already
research
seen,
is so that it
and
can be
the
consumed and
company is
applied
also
to
catching
everyday
up on
life.
it.
Right, and I think at the other end of the spectrum, news
Cool.
channel outlets.
Good answer.
The general
Hopefully.
layman person does not
Yeah,
go
well,
and get
obviously, I
their
agree.
information from a scientific, from a relevant scientific source.
So
They
the
are not
question is,
looking at PubMed.
in
They
your
are not looking
experience,
at
I'm really curious
national
to hear your
portals of
this one, actually. So
research.
in your
They are looking
experience,
at the
like,
newspaper
taking,
outlets. They are going on social media.
Oh wait,
and
can I just
they
add?
are
I'm
researching.
gonna add
The
another
thing is,
thing.
things can be
So I
very
feel
easily
like
taken
it's
out of
on
context.
underrated.
And on
It's like
social
I'm
media,
very
there is no
production
liability
oriented,
to
so
be
I think it's
credible
like
with your sources of
monitoring.
information. So it's very
So like
easy to misinterpret and misanalyze what is being put forward. But yes, I do believe that we as scientists can still do a better job of portraying it, and there should be methods and checks in place to stop this misinformation as much much as possible. I'd also like to address something that Pranav mentioned in one of his answers, that we as a team also chose to do our study via social media.
Trying to sound smart, you know?
This was because we wanted to highlight the
I think it's
biases and the
successfully
problems
sounding
with
smart.
it. If we,
But
going through that
the
process,
second
we
question is,
realized
in your
all the
experience,
biases that exist.
taking
And the
projects
studies,
from
we have
like
already seen
training
it with the
into
results of
production deployed
studies that we
systems,
analyzed,
where's the
but we
biggest bottleneck? Like,
replicated
what's
the
the
results
hardest
ourselves.
step
Of
in
the
that?
1,200 participants that we shared it to, these were also shared by the third parties
like,
that we
really
initially
right
shared
now
it to.
is
And these were
when you have
shared
very
all
big
across the
models,
country via our
it's
network.
really
But this is the
slow
thing,
to
it's our network.
inferencing.
The 87
Inference time.
respondents
Wow,
we
cool.
did have
Yeah,
were first
yeah,
or
it's
second degree
very slow.
contacts with us.
And it can be
it
very
leads to a
costly.
huge selection bias in our supposed
for
pool
example,
of
like
participants.
if you try
And
to take a
therein comes the
GPU
problem. That is the
to go
problem.
into
Social media is
productions,
supposed to offer us
and you just
diversity
even
with
use
answers. It's
Spark
supposed to order us with
instances,
ease. But if
even
it
so
comes
they
at the
cost
cost of
quite a bit
selection bias,
for
your results
every
anyway are going to
inferencing,
be
like for every time you make a
miscrew.
prediction, like
Thank
you
you.
generate something. So that's hard. Like if you just like so yes, this is why
Yeah,
we
we can
haven't
go to
seen a
the
lot of
Praelium section.
GPT-2
Yeah.
in production yet.
So again,
Right.
a very good
And it's very
debate
interesting
and very
problem. So
interesting
like
topic.
I'm not sure
I
if
would
I
like
can
to
mention the
ask
exact company.
proposition team too
But some startup
that
told me
see
so they're using
whatever research we are doing,
GPT-2
the
in production,
importance of the research
and
is the reliability.
one
So
is,
do you think by
and they say like
choosing this
if they could
particular mode of
reduce
social media,
inference time
the
by
reliability,
half,
because
they
unless
could be able to
whatever
break even.
conclusions
So
you
like
draw from that
so like for
particular
example, if
research,
they can
you are going to use in your
instead of
day-to-day practice.
using the normal
If the reliability is
precision
less,
point,
will you be
if they
able
can
to
somehow
use that
make it work
particular
on
research? And second thing
person
is,
EP16
many times you want to
precision
do
point,
research in the
then
geriatric
it can
population,
reduce
old
inference
elderly
time by
people,
half,
and
and
in
therefore
India
will
also, if you
can,
see,
like
still
help
large
the
number
company
of
like stay afloat. And I think
elderly
it's make a
population,
really
they
big
are
difference.
not
Like you just like
computer
break
savvy, they are
even
not technology savvy
or
that
not
much.
in,
This I
especially
have
in this
seen
economy.
even in the city like Mumbai and nearby.
Yeah,
So
totally.
let us accept that they are not
Wow,
as
great
savvy
answer.
as that to
Interesting.
take these particular surveys.
My
So how do
final
you
question actually
answer
is
these
simple. So
particular
yeah, if people want
problems
to
about
like
the
learn more about your
research on
work,
social
or
media?
do you have a Twitter account or a company that you want
Yes,
to tell us about?
I spend too much time on Twitter, and
Have
I'm
you
So
ashamed.
yes, sir.
Follow
So
me on
with
Twitter.
regards to the reliability
What's your
of
Twitter,
data, that would be particularly
Is
important when you're talking of surveys over social
Chipro.
media.
It's like
However, these surveys may have
CHIPRO,
robust
and
inclusion
it's
and
not
exclusion
Chipro. It's like
criteria.
professional.
Social
It's
media
more like
would just be a
RO
point from where
means
the
crazy.
participants are
And
attracted.
so
That's
actually
one thing.
Chipro.
Oh, really?
The
I did
second
not
thing
know
is
that.
when you talk of geriatric population around
Yeah.
Awesome.
I said dental.
All right, we should edit that. That was great.
Yeah, thanks.
We would request
I
all
have a
other
blog, which
participants
I
to
blog
please
about
mute
tech,
themselves. Sushmita
but
and Ritwik, please mute
I write
yourself.
long form. Like each my blog post takes me like 2 to 3 months to
So
write.
the study conducted on
Yeah,
lung
so I
cancer
don't
among
write a lot.
chronic smokers, and that
but you should
you
check
write
it
well.
out. It is.
the
That's great.
Thank you.
participants were
Thank
recruited
you.
was
Coming
social
from you,
media,
it
and
means
the
a
mean
lot.
age of
Yeah.
recruited participants was around 63.4 years, which does delve into the geriatric population. Other than that, so the social media, when talking of reliability, in terms of dissemination of information, it is a reliable tool. Now, just as the opposing team said that misinformation does exist and people are not going to look at PubMed every day for information, and they're going to actually go to news
Okay, so
channels
good
or
morning,
social media
everyone,
just
and
for the
thanks for
sake of
coming
it.
to my
Social media
tutorial
would
on
actually
modern time
provide
series
links, can provide
analysis.
links to these articles, and
This
that
is
would
sort of
actually
my
deal with the
interpretation of modern time series
misinformation part
analysis. I
as
suppose
well.
everyone
And
would have
in
a
fact,
different interpretation,
any
but to me,
research
mainly what that
would
means
reach
is
a greater
we're not going to
mass of
cover
population than it
autoregressive
normally
models.
would
We're going to talk about
through
them
social
briefly,
media.
and then we're going to mainly talk about methods that are more computationally taxing. Some of them were
Now I
invented
would
many
like to
decades
ask the
ago,
second team,
but I would still classify them as
team
modern,
number
given that
9
they've been far
is
more
opposing this
successful sort of
particular
with the
view.
era of big data, and far
That
more successful with sort
for
of
any
readily
research to
available
be
computing
more
resources.
and more
But that doesn't
reliable,
necessarily mean the
the
ideas
sample size is
are
very
very new.
important.
But I mean,
So
that's also
sample
true
size
for
is bigger,
even neural
it
networks,
is
right?
more
The ideas
likely
are
to
also
be
not
more
new.
reliable.
A lot of the modernity
The
comes
more
from computing resources.
sample size usually
So that's
involves bigger cost.
how we'll be thinking about
And
this.
if
A
you
little
want
bit
to
about me
include
and my background in this
tens of
subject. So a
thousands
few
of
years ago, I
patients
actually
or
gave another
people,
tutorial on
then
time series
the
analysis
cost
at
of the thing
SciPy.
escalates too much.
And that one
If you
covered
use
more
social media,
exploratory data
the
analysis,
cost
data
will come down
processing,
significantly. There
and
is no doubt
also
about it that
more
the cost of
traditional
the thing will
methods
come down
like
significantly. Don't you
ARIMA
think so?
models,
And if
autoregressive
the
models, that
number
sort of
is
thing.
huge,
So if
then
you're
the
interested in
reliability
that
will
and you don't have a
automatically
background,
increase.
that
So
is
that is
one
one way of
resource
thing
of
you can
many
at it.
resources.
So
That's
what is
just
your
the one that
opinion
I've
about it?
prepared. You don't need to be familiar with that
So I
material
think
for today.
you were right. It does
but
provide
it
a
is
larger
definitely
sample size.
material
but I
you
think we
should
also
be
need to
familiar
argue
with,
what
and
the
that should really be your
deviation
bread
in
and
the
butter if you
demographic
are
for the
working with
sample
time
size
series
is.
professionally.
I think,
Even though
like
those
Sadir previously said in one
those
of his
methods are
points,
sort of low tech or
the
less modern.
general
They're very
demographic
much
that is on
relevant today. They
actively
still
on social
perform
media
exceptionally
are
well,
people
and they
who
also give you a
are in
really
the
good
younger age
conceptual
group.
foundation.
Like you yourself
And
had a question
similarly,
to
it's
them,
good
what
to
are you going to
be
do about
comfortable with
larger
data
age
cleaning of
groups? And they
time
quoted a
series and all
study
of that.
for chronic smokers and
So that's a
lung
little bit
cancer.
about me.
I think they only
I'm
mentioned
also
the
just a
mean
little bit
age, which was
curious about
63.4 years.
people in this
But
room,
actually,
so I guess
the mean
I'll
of
just do a
a
quick
data set
survey.
does not
Show of
matter
hands
if you do not mention the
already work with
standard
time
deviation
series
between the
data
data
at work.
set. So if there was only one
Okay.
outlier person who
I
was
hope there will
adapted
still
to
be
using
stuff for
social
you.
media,
If
who's
not,
at the age of
no
80
hard
years,
feelings.
that would still
You can
bring
certainly
the
pipe in
mean
with your
much
own
closer to an older age.
suggestions and input as well.
And when it comes to the cost
And
effectiveness of
how
a
many
study, yes,
people
I
have
do believe
never worked
social
with
media
time
would
series
really
data?
help bring down costs for conducting
Okay.
a very large
very
scale
limited.
study.
How about very
But then
limited?
we also have to argue the cost
Okay, fair
beneficence relationship.
And how many
If we
people
are
work
not
in
getting,
a
if we are
scientific
even if
sector,
we're getting a
broadly
larger
defined?
sample size with no
Okay, and how many people
deviations,
don't work in
it's
something
not
you would
really
call
a
remotely
true
scientific?
representation of the population at large. So the cost beneficence
Good
relationship
for
is
me to
going to
know.
be
And then just out
very
of
skewed
curiosity,
towards
who
a
thinks they
certain
came the
demographic of the
farthest to get
population
here?
and not true for the entire population.
Can anybody beat Singapore?
Okay, thank you
That's
so
really
much.
tough.
Sir, if you would allow me to add.
Yeah, please.
Yeah, that's okay. There was
Sir,
someone on
the
my
point
flight
that you
who claimed they'd
mentioned
been
about the
traveling for
cost
2
being,
days, but they didn't come
reduced
to this
what with
conference.
the
They
use
might
of
have
social
you beat.
media. This was something.
far as,
This
like,
is a line
time
that
goes.
another judge
Okay.
at a recent debate told me in stage
So
in front
here's
of
an
everyone. I
outline
had
of
no
what
answer
we're going to
for
be
it.
talking
He
about.
said
Just a
quality
very brief
comes at a cost.
overview of sort of what makes time
And therefore, the
series data
cost benefit
special,
ratio
some
is
things you
something
want
that
to
we
keep in your
definitely
head
must consider.
when you're looking
If
at time series data,
and
especially if
as
you're
we
new to it or
demonstrated
haven't
in our
worked with
study,
it
as
for
we
a
have
while.
told, we
And
could
then we're going to
replicate a
divide our
larger study
discussion
on a
into
smaller
3
scale is sometimes
components.
demonstrated.
We're
That
going to be looking at
link out or
state-space
sending a message out
models
to a
for time
larger
series, just
demographic
an
of
overview of
population,
what those
larger
are,
demographic,
and we're going
does
to
not
cover
result in
2
significantly
methods
more
specific
responses
to
to the study itself.
state-space methods.
There was another example
We're going to talk
by
a
again
bit
Dr.
about
Chittaranjan
applying
Hamzade, where he
traditional,
said that sending out a
machine
study on
learning
social media
methods,
is like printing
specifically
out
for
a
time
stack of
series
questionnaires,
and
leaving it
what
in a park
approaches you
bench
can
and letting
take
the
there
wind
and
take
what
it
you want to
wherever
look
it
out
goes.
for.
And then
And
most
we're
of the
going to
people
wrap
that
up
see
looking
this
at
paper
some
are
deep
going to
learning
think
for
it's
time
trash, it's
series.
litter, they're going to throw it in a dustbin. Some of them will
Time
pick it up, they'll
series
still throw it anyway, they don't
What
want to
are time
respond.
series? It
Very
seems
few
like
will actually
everybody
go on and
has a
respond to the
good
survey,
idea of
and
what
then
those
we
are,
will,
given
then
that
we
you
can
work with them, right?
extract the data that we
The
acquire
classic
from here.
example, I think, in every
And
time
besides,
series
this
book,
is something in
almost always
response to
you
the
will run
population,
into
it's
many,
very
many
small.
examples
When you
of
say that
something like
social media
stock
can do
prices.
XYZ, then
They are
social
readily
media can
available,
provide links.
usually
Let us not forget the
free,
massive scandal
fairly
that happened
chaotic
in the
time
United
series,
States
so they're really
when
interesting to look
Mark
at.
Zuckerberg,
And
the
of course,
CEO
nobody
of
is all that
Facebook,
good
was
at
questioned
modeling
by
them because if they were, they'd be filthy rich. So it remains sort of an open
about
problem,
false
which is also
information being
interesting.
spread on Facebook, about
But I want to
misinformation
point
and
out
politically
that
inclined
many
information
other
being
things are
shared on
time
Facebook,
series
which
that don't get
Facebook took no
called
action
time
to
series and
censor
even get
fakes. Facebook took
analyzed
no
in their
action to
respective
fact check.
disciplines without
And this was
anyone
believed
using
to have
that
heavily
word, even
skewed
though
the
that's
result
what they
of the
are. So on
American
the upper right-hand corner of this
presidential elections
slide,
at the time.
I have an
And
EKG
therefore,
diagram.
this point is absolutely
I've never
moot
heard a
that
doctor
social
talk
media can in
about
fact
time
provide you
series
with good
data, but
quality
actually
and
that's
highly
often
filtered
what they're
information.
looking at, right?
Thank you.
They are taking some sort of measurement over time. When you're hooked up to any device in the hospital, it has some kind of digital or analog curve. That's a time series, right?
Now we'll quickly sum up the debate
I think
with the
data
closing
science
speeches.
or time
We would
series
request you
analysis
all
or
to
whatever
make
you
a
want to
closing
call
speech
it
of
has
just
a lot
1
to
minute and do
contribute
not
to
go
this field.
overtime.
And while we keep
The
hearing about
proposition team can start
the
with their
benefits of healthcare for time for data
hello,
science or
so
deep learning, we
I'll
don't hear about
begin
it so
now.
much in the
So
time
I'd like to
series
reiterate
context,
that
even
with
though there's
a
actually a
growing
lot
number
of
of
time
studies
series data in
being
the health
called up
field,
due
as well
to
as many others.
financial concerns and
Similarly,
recruitment
on the
constraints,
bottom,
social
I
media
think
is an
this is an
essential tool
NMR,
that
but I
allows
might be
for
wrong. If a chemist wants
cost-effective
to correct me
recruitment and
and
quick
tell me this is some
recruitment.
other kind of
Now,
spectroscopy,
there are
but this is some some kind of
with-
NMR
again,
looking
sorry.
at molecular
Facebook also
structure,
has
right?
2.9 billion
This is
users
also a
currently, and Twitter
time
has 450
series by
million
my
active users.
definition, even
Now,
though
through the use of
here
Twitter
the
alone, it was found that,
x-axis is not
the
time. It's actually just
citations in
wavelength.
the gastrointestinal endoscopy journal showed a positive correlation
Because
to
you can use many of the
tweeting
same
about
methods
the
on
research
it,
findings,
and what you
and
have is you
also-
have a well-ordered, well-spaced temporal
Mayo
axis.
Clinic conducted a study
So we
where
are
the
even
social
going to be
media
working with
accounts
some
of
data
use,
today
and,
where your
this too
temporal
reported,
axis is
an
not
increase
time
in
per
full
se,
text article
but it's something with
downloads
this sort of
and
well-ordered
citation-
metric
not citation,
between
full text
different
article
points in
downloads
time, right?
and,
So that's what makes time series different from
site
anything
visits.
that's cross-sectional
And,
analysis.
coming to the point
And here's
of,
an example.
certain
So
tribal people
what might I do
being,
with something like that, right? If I don't have time as
not active on
an
social-
axis, am I really
Yeah,
going to do
however, in
prediction?
case
Well,
any
probably not,
problems-
right? Prediction might not be the most useful thing, although maybe you can contrive an
The
example.
next team,
But But you can certainly do something like time series clustering, right? You might have multiple NMR
Ladies,
spectra
gentlemen,
and
and
you
my
want to
fellows
find a way
who
to
do not
classify
associate
them
with
without
these gender,
having
but
hundreds of
non-binary
grad
folk.
students slaving
What we
over
have
it,
seen
right? If you
here
can
is
find
an
a way to
is
replace
an
those
attempt
grad
by the
students,
proposition
even
to
better.
malign this topic, to
Okay,
mislead
so
you, and
tasks
to
for
attempt to
time
draw
series
these false
analysis. What are
equivalencies with
things
data that
people
has been
do?
taken
Well,
from places
as
that
usual,
cannot possibly be
visualization
applied
and
to the
exploratory
population
data
by and large
analysis.
in the country that we live in.
When you do this for time
Therefore,
series,
I would
often you
not
have
like to make any
specific
new points here.
time-related
I have
temporal
already said that I
questions,
have
such as
everything. I
understanding
have
the
already said
temporal
everything that I
behavior
want
of the
to say.
data,
And the original statement
especially if your
standing that
time
social media is
axis
absolutely
is
not
actually a
crucial
time
for
axis, such as
research engagement.
seasonality, right?
I would also
Is there
like
some
to add
kind
a
of
supplementary line to that
recurrence
now,
in
in
your
fact,
data,
that
some
the
kind
quality,
of
the type of engagement that you might get from
seasonality?
social media
You can
itself
also look
might
for
be detrimental to
cyclical
the
data,
quality
not
of your
quite
research.
the same
Thank you
as
very much.
seasonal data, right? So cyclical data
Thank you for the
will have
debate.
some sort of
Now we would be
recurrence,
moving
but
on
it
to
can be
the third
damped,
debate.
and it can
The
also sort
topic
of
for the
change
third
its
debate
period
is
over time. So an
pharmacogenetic
example
testing
of
should be
seasonal
incorporated into
would
routine
be
clinical
something like
practice.
the weather, and an
Speaking
example
for the
of
motion, we have Team 7 with Tanvi
an
Pinto and
example of
Srividya.
cyclical would be
And
something like
speaking
people
against the
hypothesize
motion,
there's
we
a
have
business
Team
cycle,
10
right? Some sort of
with
boom and
Tejasvi
bust
Patil
cycle in
and
banking
Piyush
or stock
Vinchuk.
prices, that sort of thing.
So the
Um,
now the
and what's another
proposition
thing
team
we
can
usually want
start
to
with
do
their
is sort of
opening
identify underlying distributions and the nature of the temporal processes producing the
Hello?
data, right? We want to get a sense of,
Am I audible?
I have a
Yes, you're
hypothesis
audible. You can
that
give your introduction
I
and
see
then
this sort of river level
Hi,
over
good
time,
evening,
and I want to get at
Julie
sort of
and my
what
worthy
describes that
opponents. I'm
river level more
Srividya,
fundamentally.
representing Team
That's another
7,
thing I might want to
and
do,
I will
not
be
just
speaking
predict,
for the motion.
but also have a way of
Pharmacogenetic testing
describing the
should be
dynamics.
incorporated into routine clinical practice.
What else do we need to do? Well,
Can I
we can do
start?
estimation of past, present, and
So you can
future
start.
values, right? So there's also
For
this
decades,
distinction
we have
between,
known
say,
that
something like
inter-individual
filtering
variability
and
can
forecasting,
occur in
or
the
also
clinical response
smoothing.
to a drug
We'll talk
treatment.
about
The proportion of patients who respond positively to their medications is only approximately 50%, ranging from a 25 to 60%, implying that the rest of the patient population is not receiving the proper medication or is suffering from a significant therapeutic delay by switching from one medication to another until appreciable clinical benefit is attained. Around 0.08% of Indian population suffer fatal adverse drug reactions on an average. What is the solution? Pharmacogenetic testing. Let me give you a quick example to substantiate my stance. Erlotinib and gefitinib are drugs that have been on the market for several years and are used for the treatment of lung cancer. The response to gefitinib is almost entirely limited to EFGR mutation positive group, whereas mutation negative patients benefited from chemotherapy. With this information, we know exactly which patients to give what. In patients belonging to Asian population taking carbamazepine, it is recommended that testing for HLA-B1502 due to estimate a 10-fold increase in risk of Stephen-Johnson syndrome in this population. A simple information that can prevent a fatal side effect. I can go on with more examples, but these are not mere studies. These are promises to a whole new world of personalized treatments, which is more efficient, has fewer side effects, and takes lesser delays to get to the right treatment. Even on prevention aspect, early DNA testing can become key in disease prevention strategies. Since genetic information is accessible long before the disease process begins. On a larger picture, genetic testing provides an enormous database, opening a whole new horizon for research opportunities. All this with just one-time investment.
So your time is over now.
Yes.
We will move on to the opening statement of the opposition team. You can begin with your introduction.
Okay, so good evening, everyone present. I'm Tejasvi Patil, third minor MBBS
Tejasvi, 1 minute. Please, could you change your background and make it to conference? And even your partner, even yours is not there. Could you guys just do that?
Is it happening?
No, it's not. If you can, it would be great. Otherwise, okay, no, it's not happening. Then we'll just continue.
Okay.
You have to choose your background in the video setting.
But I did. Actually, I think the internet connection is problematic.
Okay, I guess that is fine. We can continue with the debate.
Is it fine?
We cannot see the background, but if there is some issue right now, we don't have the time. We shall continue with the debate.
Okay, so I'll start from the beginning. Is the introduction part included in the time limit?
No, that's not included. So you can give your introduction and then we'll start the time.
Yeah, sure. Thank you. So good evening, everyone. I'm Tejasvi Patil, third minor MBBS student from St. GS Medical College and Praelium Hospital, Mumbai. I will be speaking against the stance pharmacogenetic testing should be incorporated into routine clinical practice. I'll start. In an article published in the International Journal of Medical Practice, we found that in a developing economy like India, pharmacogenetic testing, which costs from 3,000 to 70,000 rupees, it's difficult to be implemented and it's rarely done in practice. Leandrew states that there is lack of research on pharmacogenetics, mainly of randomized controlled trials, which are a necessity, as we can't ethically predispose the population to potential drugs with unknown effects. Also, these studies need a huge set of resources, including time and money, which can be devoted to higher benefit risk ratio and high utility researches. An article entitled Pharmacogenomics, the Genetics of Variable Drug Response states use of pharmacogenetic drugs shows phenotypic variations and hence outcomes can vary even after pharmacogenetic testing is done, thus proving no significant benefit at a population level. According to Indian Journal of Pharmacology, there is lack of knowledge, awareness, and skill amongst doctors, medical students, pharmacists, and laboratory technicians for routine pharmacogenetic testing. There is lack of patient counseling in this aspect and lack of public awareness as well. Murarka et al. States expensive and advanced equipments are required for pharmacogenetic testing, and such infrastructure is present only in limited urban centers like Mumbai, Bangalore, Kolkata, Delhi, which cater only up to 7.8% of the total Indian population. According to John A. Robertson et al., such tests reveal DNA-based personal information that could be used adversely to a patient's interests. Informed consent and privacy of pharmacogenetic test results, which is very essential, hasn't been studied for clinical use of pharmacogenetic drugs. In an article on clinical pharmacology, we found that pharmacogenetic tests have very low feasibility in the public healthcare in India, where patients require prompt treatment, as a high percentage of population survives on daily wages, and where 1/4 of the population treats itself by over-the-counter drugs. In a tertiary hospital in Mumbai, we found that pharmacogenetic testing is done only for drugs like azathioprine, clopidogrel, warfarin, and allopurinol. It's done very rarely as drugs are prescribed by trial and error method. And the clinical pharmacology
Your time is over.
10 to 15 patients weekly with the cost of test being 500 rupees per patient. Thank you.
The opposition team can start with their opening statement.
The question answer round, right?
No, sir, you're not audible. Hello? You're not audible.
Okay.
Ma'am, am I audible now?
Yes, sir. Yes, sir.
Yeah. So I need to ask the opposition team regarding the first question, which I would like to ask the opposition team is that they mentioned about the ADRs. Now they told that 0.08% of the ADRs were severe enough. Am I correct? So from which side you have taken this data?
This is your opening speech.
Hi.
So
Hello, am I audible? I just wanted to clarify, is this the opening speech? Because isn't that what they just made? This is the question, right?
Sorry.
Hello?
Sorry, my bad. You can start with the question.
We can start with the question.
No, now you get 3 minutes to answer the questions proposed by the opposition team.
Okay. So the prevalence of, this was taken from the prevalence of ADR in India, which was conducted in 2009, I'm sorry. And this was taken from the Indian Journal of Community Medicine.
So if you look into the recent data, which was like, if you go to the pharmacovigilance program of India, which is under
sorry, but cross questioning is not allowed.
Okay. So then the another question which I need to pose to the opposition team is that they told about one-time investment. So what amount is required? Do you know that what is the cost of the machines which is used for genomic testing? And what is the cost of each test in the private laboratories?
If you have any other questions, could you ask them together? Because the questions are to be asked together and the answers are together. So that's...
Okay. So one thing about the one-time investment, what is the cost of the machine and the infrastructure? The second is about the cost of the test which is done in the private labs. And the third is about the clinical utility. That is, what is the current evidence of clinical utility of the pharmacogenetics? The fourth question is about the healthcare utilization, that whether doing pharmacogenetic testing has reduced the number of patients coming to the hospital or not. So these are the 4 questions I need to.
Okay. In response to the questions that you just gave us, so to state an article that was given in the American Society of Clinical Pharmacology journal, it clearly states that according to an economic evaluation by the FDA, so they just estimated testing of CYP to C9 gene and in relation to initializing warfarin therapy. And as you know, warfarin is used for anticoagulation. So when they studied that, they realized that the economy could save 1.1 billion on just screening everyone for what gene makeup they have in respect to this gene. And because that results in avoiding almost 85,000 serious bleeding complications and 17,000 stroke cases each year. So while it seems like the investment is a huge amount and may not, the cost effectiveness of pharmacogenetic testing is exactly what you're asking about. So while it seems like this seems like a huge investment and a big amount, and it may not prove to be as effective, it is according to this study. And to go on to further quote the study that my teammate mentioned before, even the HLA-B1502 gene, while used for treatment in carbamazepine and Stevens-Johnson syndrome, it clearly states that the country, for every 222 patients screened, so that's not a big patient load, a big number.
Your time is over.
Okay.
Now you can start asking your question and then the opposition team can answer.
Team 7 gets 1 minute to ask the questions to the opposition team.
While you stated numerous studies that quoted the limitations of using pharmacogenetic testing and the cost and the availability, however, it came to my attention that you were highlighting the need for something else. If not pharmacogenetic testing and individualization of medications, what do you think could be the other possible outcome to provide better treatment, since that is the end goal?
That's all. This is the only question, or any more to add?
That's it.
Okay. So can we start answering?
Yes, you have 3 minutes to answer the questions.
Okay, so as we know that our ultimate goal is to give better treatment to the patients. So it is not the personalizing of the medicine which will solve the problem. Rather, it is first thing, and the very ground level thing is availability of the medicines. First thing, that is the availability. Second is the cost of the medication. That is the generic medications which cost less as compared to the brand name medications. So that should be readily available. Third could be that the physician who is prescribing the medicines should counsel the patient regarding the ADRs which can happen. So I think that these things, that ADR, counseling the patient about the ADRs, and then the readily available medicines, and then reducing the cost of the medications, can lead to a better patient outcome rather than going to genomics and personalizing the medicines. If my colleague want to add in this, it would be great.
Okay, so I was basically highlighting the thing that in a developing economy like India, where people are not getting any basic medical facilities, pharmacogenetic testing is basically not feasible. So the thing here is about the economical aspect more. Also, the infrastructure is not available. The equipments are very expensive. We took a survey in a tertiary hospital in Mumbai, in KM, and the clinical pharmacology department stated that they don't have the infrastructure, nor they have the equipment, and they hardly find like 15 patients per week who can be treated. So there is no public awareness and there is no knowledge amongst the doctors themselves about pharmacogenetic testing. So it's not feasible in India. So the need for pharmacogenetic testing does not arise. Thank you.
Now that we're done with the rebuttal, we'll hand it over to the judges for the judges' questions. Please note, keep your answers brief. For each answer, you will get 1 minute only.
Okay, I'll go first, and my first question would be to the team which was speaking for the motion, who wanted the integration of pharmacogenomics into the routine clinical practice, right? So I'm sorry to say that realistically speaking, we as clinicians are not adequately trained to interpret these genetic results as and when they come in detail, let alone change our therapeutics or therapy plan according to them. Now, how do you plan to circumvent this major roadblock? This is my question to the team which was speaking for the motion.
Good evening, ma'am. Thank you for that question. As you said, that the clinicians are not trained enough, but that is with any sort of new technology. For example, 15 years back, laparoscopic surgery by surgeons, well-known surgeons, was feared. But now laparoscopic surgery is not even considered as a cosmetic procedure. It is considered as a routine. So we do agree that there will be a learning curve, but we strongly feel that the learning curve is necessary. It will help patients, and eventually, once it becomes generalized and available, everyone will get better at it, like everything else in healthcare.
Okay, thank you for your response. My next question would be to the team which was speaking against the motion. This point has already been brought out, and we all know about the targeted therapies which in different malignancies, and similar targeted therapies are now coming up in most of the disorders that we are dealing with. And these targeted therapies would actually be game changers as far as the outcomes go. So we all know that we follow the principles of primum non nocere. We are not supposed to do any harm. So in case such a therapy is available and we know about it, how do you ethically justify not using pharmacogenetics and in these scenarios? I'm just talking about the ethics of it. I know you brought about the points about the resource limitation, but how do you ethically justify in medical practice?
Ma'am, so like ethically, like, you know, we all know that as far as the pharmacogenomics is considered, there is always a problem of data storage, of quality assurance, and of mapping. And you know, like furthermore, there is also like we need to avoid the bias of the data which we have. So there are a lot of ethical issues, the privacy issues, which are connected to pharmacogenomic testing. So like these ethical issues, and also there is no board which deals with this issue, which deals with these issues. So therefore, I think that if we do all these targeted therapies, and for that if we do all the pharmacogenomics, then definitely that data can leak, and that can have an deleterious complications.
Can I add?
Yeah.
Please go ahead.
Okay. So, ma'am, ethically speaking, firstly, like for malignancies, and it's very important that pharmacogenetic testing is done because the treatment outcome is really great there. So, firstly, we can take the consent of the patient. Second thing, we have to counsel the patient properly, like the risks of this, the benefits of this. The patient is unaware about it. So it is the responsibility of the medical fraternity to explain the patient. Third thing, the data which we have has to be kept confidential. Like it should not be misused because it's very sensitive information. So these three things of consent, counseling, and confidentiality play an important role, I feel. Thank you.
Thank you for your answer.
Okay, so this question is to the team 7, team in favor of proposition. In general, all clinicians are in favor of personalizing treatment, and public health people like me are always for standardizing treatment. Everybody gets the same thing. It makes life easier. It is easy to make an inventories. It is easy to improve the quality of treatment. But I would like to interpret this team actually just routine clinical practice, which does not necessarily mean that it has to be offered to everybody. It can still be offered to a select group only based on those who fail treatment initially. So you start with the standardized treatment and personalize only among those who is required. Even that would fit into the overall argument of routine clinical practice. But the way most of you argue, it appears as if it must be provided as a universal for everybody. You get diagnosed anything, you do a genetic testing. So I'm saying there is no need for universal genetic testing. Only still it will be a targeted genetic testing. How do you respond to that?
You can start answering the question.
Team 7.
Hello?
Yeah.
So in response to your question, what we were aiming at was better treatment for everybody. While we do understand that it will be difficult, we're not putting away the idea of standardized treatment for everybody, but we would like the importance of genetic testing. That will create a bigger database, which will help us to treat people better. For example, when we look at the treatment of hypertension, we do treat hypertension differently in different countries. And this is mainly because of the genetic makeup of people in that country. So for example, the initial drug given in the US is not the same as the initial drug that we give in India. And this is basically because of different genetic makeup. So if we study this further and if we create a database by genetic testing, we will be able to provide better healthcare, is what we feel. And from the preventive aspect as well, since we are community medicine professionals, even from the preventive aspect, we feel like if we have a genetic testing, we can target disease processes even before they start. And hopefully someday we can prevent conditions like cancer if there is a genetic susceptibility. Thank you.
Okay, thank you. I don't think the treatment of hypertension is different between countries because of genetic makeup, but anyhow, we can, that's all for you. Now I go to team number 10. See, largely your concerns of doing pharmacogenetic testing relates to cost and capacity. But we know from our enormous experience that the moment you make it available to everybody, the cost comes down drastically. They come down by 100 times. Something which costs 10,000, 50,000 becomes 200, 300 in a very rapid pace of time. And you can also easily convert them into point of care districts which are again cheaper. So today technology exists that these things can really become very cheap and be done as a point of care district. So cost and capacity to me are not the major barriers. How do you respond to that?
So I think we may have focused on the cost, but it is not just about the cost. It is rather a systematic review which was published in Pharmacogenomics Journal March.
I can't hear you.
We're not able to hear you. Can you check your network once?
So maybe Tejasvi can start in.
Robust and pragmatic.
We aren't able to hear you very clearly.
Tejasvi, you want to answer? Because we can't hear him.
Oh, yes, sir.
Am I audible now?
So
Okay.
which one of you would be answering the question?
Piyush?
Am I audible?
So,
yes, you're audible now. You can answer.
Okay. So I was saying that although we must have focused on the economics part of it, but also if we come to the clinical utility, an article which was published in just March 2022, that is just 3 to 4 months back, it also stated, which is a systematic review, it stated that although the pharmacogenetic can individualize therapy, but we need more studies, more randomized controlled trials in order to establish the impact of the pharmacogenetics in a routine practice. So the clinical utility part of the pharmacogenetic testing is also in very infancy, and it is not robust evidence that it is very useful.
Sir, I would like to add to it.
Go ahead.
Yes. So also this procedure is very time consuming. So patients require prompt treatment in India, as most of them don't want to miss their working days. So the time is an important factor here. Second thing is about knowledge. The practicing clinicians, the medical students, the lab technicians, they have very limited knowledge about this. The clinicians also don't have knowledge to interpret the thing is that so adding to the point of time, the time consumption part, patients, if they don't find prompt treatment, they can go for over-the-counter drugs they go for over-the-counter drugs anytime. So that is a problem again. And infrastructure is a problem. The equipment required for these are advanced and they are very expensive. So availability in India is not that seen. So
okay, that's all from my side.
Okay, so first I would like to ask Team 7 that from where you read that in USA and India, we treat hypertension with different drugs. Because if you see the Indian guidelines and American guidelines or British guidelines, they are the same. There is no difference. So from where you read, that's my first question. Can you hear me? Yeah. From where you got this information?
Hello. There is an ethnic difference in control.
Is there? Yeah.
So that is what we were trying to say, that based on the ethnic difference, the genetic makeup is also different, and that and hence the response to drugs will also be different.
But if you see the guidelines, Indian guidelines as well as American guidelines are same. Anyway, keep that apart. My question is what's the reliability of these tests? Because in certain drugs, the pharmacogenomics, it's a developing science, right? So for few drugs, it is a good guiding thing. But there are many other drugs in which the pharmacogenetics data is not very reliable. If you go through the recently published articles, in many drugs situations, it is not reliable. So still you would like to ask the patient to undergo this. Number one, the cost, and number two is the time which is required. That is number two. The third thing is, again, like the previous debate, here also the government hospitals or government medical colleges are not doing this test to a large extent. Very few may be doing, as a KM also doing 3-4 tests, as they said. So it will not be available to large number of population in India. So what is your opinion on that?
Sir, we are hoping for a brighter future with this. That is, this initially, like 10 years or 20 years ago, the many tests that we are doing very easily, very accessible tests that we see today weren't very affordable back then. But now we cannot think of a routine clinical practice without those drugs. Without those tests. Example for like MRI, for instance. Those were considered pretty expensive once upon a time. So we feel like if this are getting incorporated, there are more researches happening on this. This, as we already mentioned, the genetic database that this opens up into a wide array of research opportunities. So if this is kept forward without fearing that this might not be very affordable to patients, or if it might not be good use of resource. Instead of that, if we keep forward, it has a promising future. In the future, we would look back and think that this was indeed successful.
Okay, thank you. So team 10, I would like to ask you that how can you say no to the technology? See, technology in the initial stage may be costly. Over a period of time, when large number of population starts using the technology, then there is improvement in the technique. There is a decrease in the disadvantages like cost and various other things like learning curve, you're saying. So you cannot say no to the technology. And if you see in Western countries, if you have a condition like acute lymphatic leukemia, they give pharmacogenomic wise personalized individualized therapy. With that, the success rate or the cure rate is more than 95%. So if that is the case, how can you say no to the technology? That is my question to you.
Sir, so talking about the routine clinical practice, sir, we see that there is pharmacogenetic variation as well associated with pharmacogenetic testing. Like suppose if we prescribe some specific drugs, they can have different effects in the individual with the same genes as well. That is because of phenotypic variation. So that is one factor which we could consider for routine clinical.
Your voice.
So, am I?
Tejasvi, we cannot.
Not audible. Yeah.
Am I audible?
Yeah.
So yes, sir. So because of the phenotypic variation seen, the benefit-risk ratio is decreasing because of that one thing. Second thing, the clinical research lacks in this field. Clinical pharmacology field in India, it's a growing field. The field still needs to expand. It needs to include medical fraternity. It needs to include the students. It needs to include the laboratory experts as well. So it's a growing field, and hence our opposition to it. And also, sir, till now, the stats in India have shown that only 7.8% of the population is benefited by it, as it is mostly only present in the urban centers. So including it in the routine clinical practice can be a bit risky. And also, if some doctors practice it and another doctors don't practice the protocol, patients will be diverted towards the doctor.
So your time limit to answer the question is over.
Okay. Okay, so thank you both the teams. Thank you.
Thank you, sir.
Thank you.
Now to sum up the debate, let's move on to the closing speeches. The proposition can start with their closing speech, followed by the opposition. You have 1 minute for each speech, so you can start with your introduction and then begin off with this.
Good evening to everyone here. I thank you all for such a wonderful evening. I am Dr. Tanvi and I will sum up our stance. So personalized medicine offers the potential to revolutionize healthcare delivery through improving the effectiveness of treatments while simultaneously reducing side effects and the inappropriate use of the scarce medical resources that we have. While we discussed in length today about how we should continue what we have been doing since ages, we are here to stand for a more promising future. Our genetic makeup holds more secrets, more clues, more potential than any doctor's checklist has found so far. The medical field should progress from relying solely on standardized treatment and step into an age where we look truly at an individual for being treated for their individual genetic makeup. Thank you.
Moving on to the closing speech of the opposition teams.
Audible?
Yes, you're audible.
So, good evening, everyone. I would like to close the statement of the debate by saying that in a developing country like India, where patients still don't have access for institutional deliveries, where they don't have access to specialized newborn care by pediatricians, and where sometimes logistic problems are such that people don't get tetanus toxoid injections in peripheral health centers and emergency ambulance services, having genetic testing, the pharmacogenomics, which is not for research area, hanging in infancy, where there is unsurety of the clinical utility, and it is also monetarily expensive, and there is no robust standard clinical guidelines, and something which is having regulatory issues, having a potential threat of leaking confidential information in routine clinical practice seems to be a far and an impractical approach. The funds and the human resources which are to be used for pharmacogenetics rather should be channelized to more ground level and emergency problems like reproductive child health care, like immunization and like strengthening the emergency ambulance service in India. Thank you.
Thank you for this debate. Now we would move on to the next debate. The topic for the next debate is, it is ethical to use research data in public domain. Speaking for the motion, we have team 4 with Nitya Vaidya and Aryan Tambe. And speaking against the motion, we have team 6 with Shirish Rao and Yash Kamath. We would start the debate with the opening speech of the proposition team. You can introduce yourself and then start.
Good evening. Am I audible?
Yes, you're
Hello?
audible. Yes,
Okay.
yes, you're audible.
Good evening, everyone. I'm Nitya Vaidya from Team 4, and I will be speaking for the motion. Can I start?
Yes, you
Hello?
can start.
Okay.
Yes, yes.
Groundbreaking research is driven by data, be it predicting potential disease outbreaks or mental health trends. The public domain provides a treasure house of population-level statistics and knowledge. The very existence of the public debate, of the public domain, is to facilitate the free transfer of information and empower researchers to derive new worth from already formulated data. I'm Nitya Vaidya, and I'm vehemently for this motion. The public domain includes that which is not protected by copyright and can be freely accessed, redistributed, and reanalyzed. Hence, it is legally acceptable to use this data as the user has by default given their implied consent. Therefore, conducting research in the public domain entails pre-filled consent of the individual, maintenance of complete anonymity, and these observational studies carry no harm to the participants. This research is performed for societal benefit and honoring all 4 pillars of bioethics. Renowned organizations like the British Psychological Society maintain that conducting research in the public domain does not breach the medical code of ethics. By claiming such data as unethical, the opposition is closing off the use of national statistics, public research papers, newspaper articles, etc. These are important sources without which no research can be conducted accurately. June Mikal et al. From the BMJ states, internet users were aware and not opposed to the use of available data for health monitoring activities, provided this data remained anonymous. While Elizabeth Hilf et al. From the BMJ concluded that it was the lack of knowledge about research methodologies that led people to deny secondary analysis of data. And once educated, most participants agreed. An important testament to the ethical standing of the public domain was when the New England Journal of Medicine made its COVID-19 papers public to facilitate global collaboration and secondary research. And after all, it's called research. In conclusion, there is no right or wrong in ethics. There is only better.
I'm sorry
and
to
worse.
interrupt, but your time is over.
Yeah. Thank you.
Okay. Now the opposition team we present their opening speech. You have 2 minutes for to present your opening statements.
Hello, am I audible?
Yes,
Yes.
you're audible.
Okay. Good evening to the chair and to the audience. Today, my colleague and I will be protesting the motion that it is ethical to use research data in the public domain. Firstly, as my esteemed opponent stated, we must understand what public domain means. It is simply the information that is available and can be utilized by each and every member of the population for their benefit or for whatever purposes they may desire. So, in simple words, we can say that this common knowledge or it is a shared commodity that belongs to everyone. And it is therefore assumed that this relieves the researcher from the burden to seek ethical approval. However, this is far from the reality. While we presume that this data is safe and anonymous and free for everyone to use without any consequences, a review from the Iran Journal of Public Health shows that it can contain identifiers and consent violations which are carried on from the primary study itself due to a lack of proper regulations. Furthermore, a data blog finds that private researchers and private sources have different ethical rules and guidelines that they need to follow. In many cases, they can even contain identifiers explicitly without the requirement to blank them out, thus creating completely different ballgame from whatever public research is about. Thirdly, we have the BMC Journal of Medical Ethics, which states that privacy breaches and algorithmic discrimination has become rampant in the age of the internet dataset. And a clear-cut example of this is the Cambridge Analytica and the Facebook scandal, in which the publicly available data of millions of users was used to manipulate election results in a country. Finally, we come to biases and censorships, which propagate prejudices and misconceptions about a particular community, race, caste, or any group that you might think of. When a primary research is done with these biases and it continues by the secondary researchers and it disseminates into the public domain finally, it just helps perpetuate whatever thoughts and beliefs we have about a particular grouping. So in conclusion, we would like to state that a lack of uniform regulations, inherent research biases, and selective censorship of particular topics ensure that research in the public domain can never be ethical. Thank you.
Now let's move on to the rebuttal. The second speaker of the opposition team can start with the question. You will get 1 minute to ask your question.
Hi. So my first question.
No, not you.
I am supposed to ask.
Sorry.
Yeah. Thank you, Shailaja. I have few questions to my opposition. So firstly, I would like to ask if my opponent means by what does my opponent mean by free transfer? Like, do they mean that there should be no scrutiny or no regulation on how this publicly available data is provided to the researchers as well as to the public? Second, are you aware of the difference between implied, the thing that you told about implied consent? So what is the difference between the blanket consent that is taken and the proper informed consent that is needs to be taken for research purposes? Lastly, I would like to state the databases that are available. So they have two types of data. There are either reports data or primary research data that are available, and there are data sets which are available. So for primary research data, there can be anonymity, but that can lead to not personal level of bias, but community or racial level of bias. Then these data sets have their own privacy issues, etc., which was pointed out by my teammates. So how do you plan to mitigate that in terms of like having proper regulations to do, provided we don't have uniform IEC regulations across the world, also provided there is no uniform
Time is up.
within the researchers at present about IEC. Thank you.
Thank you so much for your question. So to answer your first question about the free transfer, I do not mean the free uploading of public data onto the public domain. What I mean is free transfer of what is already in the public domain between one researcher and the other. To your second question, and obviously there are IT laws that protect publication of personal data onto any public platform, and these laws as of 2018 are a punishable offense. To your second question.
He was saying that. Yeah, so the second question that you asked was if it's primary data, you mentioned that it creates a community bias, but that really doesn't make a lot of sense considering that when you're collecting data from a pre-existing source, you're not individually going and collecting data from every person. So you already have pre-approved data that you're using later. So how does that create community bias at all?
And also to your question about the two data sets, there are definitely two data sets. There's a primary set, which the initial researcher will get the consent, and that is where the informed consent comes in. The primary researcher that is collecting the data from the people is the one that is getting informed consent for their study. When they publish this data onto the public domain, this data is completely anonymous because they have undergone a previous informed consent where they have said that their data will not be made public, as well as undergone ethical review where they have said that their data is not allowed to be made, it has to be anonymous. When you conduct secondary research on this, you do not need to get consent because the primary participant has already given what is known as implied consent to use their data furthermore for research on the same topic. And this has been confirmed by an article put in the British Medical Journal, which has stated that the people who are involved in the primary data set do not have any opposition to their data being used for further scrutiny and evaluation.
Now, if you're done with this round, the second speaker of the proposition team can begin with the questions.
All right. So my first question will be that you mentioned that you spoke about the sourcing of the information, but wouldn't that be the responsibility to collect the primary information, like the person who's done the primary research paper and published it? Wouldn't the ethical sourcing of information for that be the responsibility of that researcher and not as the person using information from the public domain. So that's one. Also, the IT Act makes it illegal to give out any personal information in the context that you mentioned. So that did not really add up. Also, by your logic, using national statistics itself would become illegal, whereas the government is required to publish the national statistics. So how do you go about dealing with the national statistics, which are by default published and can themselves be used by the public domain?
Are those all the questions? Are those all the questions?
Yes, that's all.
Okay. Okay, so we'll begin firstly with the point of sourcing. So yes, collecting the data is the responsibility of the primary researcher. Another thing which my teammate mentioned is blanket informed consent and informed consent. So blanket consent is everything is acceptable to the participant. Informed consent is the current procedure is acceptable to the participant. And oftentimes researchers do not mention the fact that their primary data will be used multiple times over in a public data set. And the participants are multiple times not aware of the fact that their research is being used, because this conversation has never happened. Secondly, you mentioned that IT Acts make the public revelation of private data illegal. So I would like to remind you that the topic is currently ethics, not legality. Ethics and legality are completely different things. Laws are enforceable. Ethics are behaviors which human beings must uphold in the spirit of good faith. Laws are enforceable. Laws are punishable. Ethics are not punishable. They are moral acts or virtuous acts that human beings must uphold as a result of being human. Finally, national statistics. Why would they become illegal? Point being that national statistics are generally gross collections of data. They represent means, medians, and averages, and do not include the same thing as a primary data set. A primary data set is one wherein patient has been identified as age, sex, gender, and their data has been compiled without their name. And what you mean by government data is how many people live in this area, how many women live in this area, what percentage of them are affected by a particular disease. So these are completely different data sets, and it wouldn't become illegal because the government is mandated to make them, and the government makes the laws. So how can the government contradict itself in whatever question you were asking? If you have any other questions, I'll be free to answer. Actually, one point which comes up is the BMJ article which was mentioned, that the patients trust the researchers or believe the researchers and are comfortable with their data being published again. This is just one study.
Team 6, we are done with the time. The time is up for this round. Yeah.
Moving on to the judges' questions. Ruchika ma'am, you can start with your question.
Yeah, okay. My first question would be for the team speaking for the motion. And I know this has been deliberated time and again, because a lot of times it has been brought up in the same debate only. But again, implied consent, the words you spoke, and the journal you quoted. So a lot of times, okay, implied consent does happen in a lot of situations, but while we are browsing through the public domain, a lot of times this implied consent is in the form of checkboxes, pop-ups, cookies. We're all in a hurry to just go through them so that we can get to the content that we want. Okay, and I think a lot of us, I mean, if not the majority of us, don't even read these in detail. So do you think this can be considered as implied consent? And to consider it as consent, is it actually ethical?
Thank you so much for your question, ma'am. Ma'am, I would like to say that although people do not read the terms of service, when one is putting out data onto a public platform, now this again comes into question, what comes under the domain of the public domain? Now, public domain are basically websites that one does not need to sign in to access. So when one publishes any sort of information on a website that you don't need to sign in to access, it is implied by most internet users. In fact, there was a study conducted which was published in, again, the British Medical Journal, wherein most of the people using the internet stated that they understood that it was implied that if they publish anything onto a public website, it was for the public to view. And because it's for the public to view, it is hence implied that one can conduct research on that. So yes, while we do not read the checkboxes, that would come under something that would come under social media. which are platforms where you have to sign in to access, and that does not come under the public domain. So that is where the real question of privacy comes under, because people think that they have privacy on social media because what they are publishing is probably within their friends and is considered private. However, accounts where you don't have to sign it is not considered private by either the end of the internet or the users of the internet. And that is why the consent is always implied, because
your time to
publishing it
answer
on a
the
public
question is
platform.
over.
Thank you.
Okay, thank you for your answer. My next question to the team speaking against the notion is, so I would like you to basically deliberate on the moral obligation that comes out of the use of all these research data. So basically, if I've collected some data or I come across some data which has got major public health implications, how do I justify not doing something about it? I would like you to answer that question.
Thank you for your question, ma'am. So basically, we are not against using data which is in public domain, but it needs to be used ethically, which is currently not happening. So there are 4 ways in which this data can be used with a great public significance. So first, this data needs to be de-identified. It needs to be made sure that this data was collected for the purpose of research and such that the consent was taken. If the data was not collected for the purpose of research and it was collected for the purpose of something else, then the consent differs and the re-consent has to be taken from the participants for utilization of that data if identifying information was present. Thirdly, the outcome of the results, even though anonymized, must not provide any de-identifying information to the individual or to the entire community or any racial or any thing as such. And lastly, it should not provide damage or increase risk to the population on which it was done. So provided this criteria satisfied, public domain numbers and public domain data could be of huge importance and huge relevance. But at current, the lack of knowledge of researchers of trying to get this regulation sorted and trying to do it in an ethical and a systematic manner is poor. And even there are the ethical committee regulations. So hence we are stating that it's ethical at present, unethical at present. Thank you.
Thank you for your answer.
Okay, this is Dr. Anand here. Generally, the assumption seems to be anything which is in public domain is free for all. That seems to be the assumption, which is not true. Anything which is available in public domain does not mean that everybody has free right to use it. That argument is never true. And there is nothing called as a blanket consent. The concept does not exist. Okay, now coming to the question to number, which says, which was in favor of using this data. So my key question is, very often what happens is that the consent has been given, and including to be put it on public domain for one purpose. Whereas it, just because it is available in the public domain, it gets used for some other purpose. So how do you justify that? According to you, it is allowed. It can be done. So I'm saying, how do you agree to that?
So considering researchers are publishing content into the public domain, this content that is published undergoes a primary ethical review. So when a researcher collects information from their data, from the people that they are conducting the primary study from, they go through an ethical review in which complete anonymity is made. So when you publish onto the public domain, according to the general data laws, it states that you cannot ever publish anyone's private data or private information or identifiable information onto the public domain. And hence, researchers of the primary data make sure that there is no recognizable features in their data. So hence, when this goes on to secondary consideration, there is no question of any harm being undertaken to the participants of the primary study, because their data has never been in their personal information will never be in the hands of the secondary researcher who are just analyzing the data which is within the public domain containing no identifiable features. However, there is a lack in the market because the public domain is something that is new and emerging, and there do need to be better laws to considering the primary, like giving research by the primary researcher.
Okay, so however, basically your argument is that if there's anonymization, it doesn't really matter.
Yes,
right?
Sir.
Okay, you may want to think more about it, but we're going to the second thing. Again, as I said, there are ethical guidelines in India and laws in India to address these issues. some people might do an unethical practice, does not make the whole this thing as unethical. So
yes, sir.
My point is that you can use data available in public domain ethically.
Yes, sir.
So why are you saying
why
are you saying that this is for the other team, team 6? Why is team 6 saying that it is unethical to use data in public data? I'm saying it is ethical. Some people do unethical practice, does not make the whole thing unethical.
Okay, thank you for your question, sir. So I would like to quote some statistics from the observational studies done on the knowledge and practice of research ethics among Indian medical professionals as well as medical students. So 76% of Indian medical, 76% of participants had no proper ethics as 40% of them were not aware of the population. 68% of them believe that like sending a proposal for ethical review delays the research, hence they avoid sending the proposal for ethical review. And 96% believe that it's okay to fabricate the data that they already have. Again, this data was again validated in a larger sample of more than 1,500 participants, which states that the knowledge of ethics was poor, such that only 8 to 35% of the people were aware of the current ethical guidelines. Also, one more study of plagiarism, and so as my other participant quoted that the people are aware of how to use the primary data. There is such a poor knowledge of plagiarism in India that the average knowledge score out of 10 is 4.5 plus minus 1.7. So people don't know how to utilize the primary data to generate a secondary evidence properly. So it's being treated as unethical.
Okay. I am not convinced, but anyhow, Dr. Ajay Daksha, please.
Okay, so first to the team 4, which is for this particular motion, that the main issue which is coming here is the ethical issue. And therefore, considering this as a very important thing, the consent and all, so what kind of research you think, should we classify the research into which kind of research can be given safely in the public domain and which we have to restrict? So do you have some thoughts on this?
So definitely there are two checkpoints at this stage. One is the entry into the public domain, what kind of research can come in, and the second is what kind of research can you conduct secondary research on. So the primary research that can come into the public domain, researchers, there should be proper laws to govern this, as well as the good clinical practice certification of the research of the primary researchers that do, and the primary investigators that do undergo training for all these purposes. However, the researchers should make sure that there is no identifiable data of the participants, as well as no data that can be procured from the internet. For example, if you take a group of participants from a particular website, suppose, and then later on, when you cross-reference the articles, you can find out from which website you've taken the participants from. That should definitely not be allowed. But as Sir also mentioned, just because a system can potentially be unethical doesn't make the entire system itself unethical. The public domain exists, and with the proper use of the public domain, it is definitely an ethical system, especially with researchers going into it with a clear state of mind, saying that this is our topmost priority is to maintain the anonymity of the users, as well as maintain as in our primary data set, as well as our secondary data set, as well as go ahead with conducting research that takes the primary data set and adds more beneficial value to it. So that is one of the beneficence is one of the important pillars of bioethics. The secondary data that you collect should be beneficial to the primary participants.
Your time is over.
Okay, so thank you. The question to team number 6 is, ultimate aim of any research is improve our knowledge so that for overall benefit of the mankind. So in that case, whether the research is coming in public domain or not coming in public domain, if the laws are improved, okay, as far as the privacy and other things are concerned, why we should oppose the research coming in the public domain? So what is your views on that? The ultimate goal is to spread knowledge and improve our existing knowledge.
Yes, sir. Thank you, sir, for your question. I'll begin. So primarily, as you said, the ultimate aim of research is to improve knowledge and to benefit the world. But in the past, there have been multiple cases where there have been misrepresented research, where studies were conducted in such a way so that they distort the values and present some other result beneficial for some pharma company or for some food tech company. And this was many times the flip-flop which we saw on cholesterol. Cholesterol was good for you, cholesterol was bad for you. That is a result of such unethical practices where research numbers were distorted to benefit a particular individual or community or group. Coming to why research should be in public domain. Yes, sir, we agree that research data should be in the public domain, but at present, there are no ethical guidelines in this regard. There are legal guidelines, and legal guidelines and ethical guidelines are completely different. As mentioned before, laws can punish a person. Ethics can uphold a person to certain moral values. So there are ICMR ethics guidelines, but as mentioned by my fellow participant, they are not properly enforced, and 90% plus people do not believe in them and do not go to the IEC or to the Institutional Review Board for conducting their research. So if the primary dataset by itself is not ethically approved, how can the secondary analysis become ethically approved automatically? That the participant themselves do not know what type of study has been done on them. Secondly, what was stated was that research is expected to be beneficial. Secondary research is expected to be beneficial, but who upholds this quality? Who checks for malintention? Who checks for bad thoughts or bad intentions of the researchers towards particular participants or particular countries? And India has, of course, been a target of many such Western studies. India has been represented as a poor, backward healthcare country multiple times.
Okay, thank you.
Moving on to the closing speeches, we'll begin with the closing speech of the proposition team, followed by the opposition. You have 1 minute for this speech. You are not heard very clearly. Could you?
Hello, am I audible?
Yes, you're audible.
So in a world that is ever evolving, the only true growth comes with moving forward. However, to move forward, we must learn. To learn, we must collect, collate, and analyze. And none of this would be possible without the use of any and every source of information that we can ethically get our hands on. Using the public domain provides us with a legal yet effective way to gather large quantities of information that we can comprehend, compare, and then utilize. Using information that is in the public domain is not only ethical, but now it's also of utmost importance because it helps us create a larger database that we can then use forward. For example, if Einstein hadn't publicized his theory of relativity, if Alexander Fleming hadn't published his work on antibiotics, and most recently, if COVID-related information hadn't been made available to us, would we really have been in the same place in terms of research and development that we are today? I'd also like to say that using information from the public domain has helped us learn. It has helped us grow, and that is precisely why it always was, is, and will be the vital cog that not only stands on the pillars of bioethics, but also what makes research what it is. Thank you so much.
The opposition team can start with a closing statement.
Okay. I am Shirish, and I'll be giving the closing statement. I would like to start by stating that I am sorry to state that, but my opposition has really poorly understood the concept of publicly available data. Presently, what we are talking against about in terms of ethics is the data sets which are available publicly and not the published papers. So the problem with data sets which are available publicly is the privacy issues, and even if they are available anonymously, it can lead to grouping bias. So to avoid that, what needs to be done is we need to reform our ethical regulations such that it goes right from the education and training of researchers to education and training and better guidelines and institutional ethics committee. At present, there are no standard institutional committee guidelines across the world to regulate such studies, because of which these studies are bypassing institutional ethics committee and leading to consequences which my colleagues have already told, like negative consequences in which it has caused damage which needs to be avoided. So at present, we highly advocate that public data needs to be used for a good purpose, but at the same time, it needs to be used ethically, which at present is lacking and needs reform and will surely happen in future. Thank you so much.
It was wonderful to see this debate. Now we will be moving on to the next debate. So the last debate for today, the topic is induced pluripotent stem cells could replace embryonic stem cells in clinical application and disease remodeling. Speaking for the motion, we have team 5 with Nishit Chatnani and Tanya Jacinto. And speaking against the motion, we have team 8 with Sushmita Gupta and Ritvik Sarkar. we would request the proposition team to start with their opening statement.
Hello, I'm Nishit Chatnani. I'm from second year MBBS, and I'll be going with the opening speech. So my speech starts now. A father waiting at a hospital bed for a liver transplant, which is never going to happen. A daughter who will never be able to see herself in the mirror after an acid attack. But all of this can change with stem cells. A novel way of creating artificial stem cells was successfully done in 2006 by Shinya Yamanaka at the Frontier Medical Sciences Institute in Kyoto, Japan. They introduced 4 transcription factors into mouse fibroblast cells using a retroviral transduction mechanism and obtained pluripotent cells. Hence the name induced pluripotent stem cells, or iPSCs. From organ transplantation to tissue repair and regeneration, iPSCs have widespread clinical applications. According to a paper published in the Canadian Journal of Cardiology, for a person with end-stage heart failure, iPSCs could serve as an alternative to transplantation, reducing the need for allografts. Another application for iPSCs is creating and studying disease remodels. For example, many current therapies of Parkinson's disease only address the symptoms and not the underlying neurodegeneration of PD. Recent advances in the field of cellular reprogramming and personalized medicine now allow for previously unattainable cellular therapies and patient-specific modeling of PD using iPSCs, as mentioned in a review article by researchers at Bozeman, US. The use of autologous stem cells for cardiovascular medicine has a major advantage over ESCs, as they are patient-specific and the transplanted cells will not be identified as foreign bodies, removing the need for an immunosuppressive therapy. As stated in a letter to the editor published in the American Journal of Respiratory and Critical Care, using ESCs is the direct destruction of innocent human life. Consent for an incompetent research subject by a legal surrogate is allowed only if there is a minimal risk and therapeutic benefit for the incompetent research subject. The principle is upheld by the Declaration of Helsinki.
I'm sorry to interrupt, Nishit, but
the
your
United
time is
Nations
up.
Declaration of Human Rights. Despite the barriers to
Your time is
iPSCs
up.
research,
Yeah. The opposition team can start with their opening statements. Team 8. Team 8, you're not audible.
Hello, am I audible now?
Yes, you're audible now. You can start with your opening statements. You have 2 minutes to give your opening statements.
Thank you. Hello and good evening to all. Myself, Rutvik Sakarkar, and I will be speaking against the topic. I would like to start with a small example. Just imagine a father with his son, and the son is completely imagining and pursuing his career and willing to pursue his career in engineering. But the father is willing and completely forcing him not to do it in engineering, just be a medico and study medicine. This is what induced pluripotent stem cells actually are. The cells which are used for different purposes like squamous cell epithelium or other cell epithelium which are non-germinal epitheliums, that cell are forcefully, their genetic material are forcefully used for expression of genes which are not in their forte. So this is what actually happens in induced, let me make it very clear, induced pluripotent stem cells. So other thing is, eventually what will happen is the research which have shown there is huge number of genetic instability and there are various genetic issues such as variation in copy number. There are various lesions found in the cancer programming genes such as 14, 21 and 22. These genes have occurred in induced pluripotent stem cell. Eventually, I agree that at this point of time, at present, it will help the individual to grow better and to make his life comfortable. But what after a certain period of time? This human civilization will definitely collect a gene pool of inaccurate genes which are defective, and this will not only affect our civilization, it will affect the evolution process of ours, it will affect the health of ours in the coming future, and might even lead to extinction of human species, just because these defective genes get accumulated into the human gene pool. And this is what we are talking about, and we are strongly opposing the point that induced pluripotent stem cells should not be used. Next thing, the research, what my friend just mentioned, the same
I'm
research.
sorry, Ritvik, the time is up. Time is up.
Thank you.
Moving on to the rebuttal, the opposition team, the second speaker of the opposition team can start with the question. You get 1 minute.
Hello, dear my worthy opponent. I would like to ask you, you've mentioned a lot about how the iPSC is beneficial in regeneration and a lot of such diseases. I'd like to ask you how ESC is any less than that in iPSC, and especially you mentioned Parkinson's disease, where there is clear data stating that the neural progeny at iPSC cells is comparatively a lot more variable and low than ESC.
Oh, am I audible?
Yes, Nishit, you're audible.
Yes. So I spoke about the various benefits of induced pluripotent stem cells. And the issue over here is for embryonic stem cells, we need to take the cells from the embryos which are present in IVF clinics. So that's the biggest source of embryonic stem cells. For induced stem cells, on the other hand, we prefer a more ethical approach of making these cells in the labs ourselves. So we don't have to rely on a shaky ethical ground and take the cells from the embryos. So that is why that's the major advantage that induced have.
And I would like to add to what my partner said. So in saying that it's more beneficial in diseases like Parkinson's, when we go to study the pathology of disease, since we are going to be taking the cell from the individual who we are going to study, we have the exact genetic composition, the exact epigenetic composition, as well as intracellular composition. So it provides a better understanding of studying the pathology of disease. It helps with better understanding of remodeling of diseases. Additionally, induced pluripotent stem cells are more ethical than embryonic stem cells. So we are helping overcome the ethical dilemma that embryonic stem cells pose.
Are we allowed to rebut this?
No, cross questioning is allowed. We would be moving on now, so the proposition team can ask their question. The second speaker of the proposition team can start asking the question.
Hello,
you get
am
1
I
minute.
audible?
Yes, you're audible. You can start.
My question for the opposition team is, embryonic stem cells have been around since the 1980s. So that's almost 30 years. However, they aren't being used practically for clinical treatment due to ethical reasons and inability to be patient-specific as seen in Parkinson's. Can you account for the ethics, practical drawbacks, and cost of this procedure?
I would like to answer this question from my opponent. First of all, talking about the iPSCs, these are also in their initial stages, and this would definitely take very long to make into use. So instead of instead of giving time for this research purpose, why don't we preserve the stem cell right from now of the individual which are being taking birth right now? So this will help them to use this stem cell of their own, which were preserved from 30 years back, and then at 30 years later they can use these cells. So there won't be even need of embryonic stem cell or iPSCs at that time.
Also, I'd like to add here that the fact that ethics is being pushed into this entire area. As your partner itself mentioned that it's mostly from the IVF centers. These embryos are anyways not going to be actually useful anyway. So actually the point of ethics is something that needs to be addressed correctly. We are not really killing someone's blastocysts inside the body of a mother. We are using something that is anyways going to go waste. And I like to point out that when we're talking about ethics, there are so many things that we are not taking into consideration. There are so many, fetuses that are being aborted daily, so many mothers who are abandoning their children. And if someone is willing to donate their embryos, they should be allowed to do so. Other than that, the practical drawbacks, it's because this is something that is very complicated. Be it ESC or IPSC, both of them are extremely complicated and difficult to go through with. So I think this question is still there for both of the topics, and it will take time because we do not want to do something that will harm the humans. Thank you.
I think we're done with the rebuttal. We'll begin with the judges round. Each team gets only 1 minute to answer to every question, so make your answers brief. Ruchika ma'am, you could start with the question.
Yeah, I think we saved the most technical topic for the last. And I would like to first congratulate both the teams because I know this topic was very, very technical. Okay, so my question for the team which was speaking for the motion, right? So the debate was about whether these induced pluripotent stem cells should replace the embryonic stem cells in clinical application. Okay, so I wish you would have harped a little bit more about the should replace part. Okay, I know both of you guys, both the teams actually spoke about how the use is virtually non-existent in the present scenario. So the question is actually very far-fetched in the future where iPSCs would replace ESCs. But that aside, I'm sure you guys would have read up about the limitations of iPSCs, okay, about the availability, the cost factors, equipment required, the standardization of procedures, and the limited beneficial qualities. Like it can't be used for all diseases. Plus, there are associated adverse events like it increases the risk of a lot of malignancies. There are genetic mutations that happen. How do you defend the use of iPSCs then? Okay, the list of adverse events is the list of limitations is actually too huge for me to rattle out right now. I'm sure you've read about them.
We
So just
actually lost
justify.
your audio in between, so we didn't hear a major part of it. Could you please repeat?
So my question is, I'm sure you guys would have read up about the limitations of iPSCs. Okay, my first question is the debate itself. iPSCs should replace. Do you think that is practically feasible? And the second part of it, the limitations about the availability, the cost factor, the equipments, and the adverse effects which are associated with iPSCs and not there in EPSCs, in the ESCs. How do you defend that? Could you get me this time?
Team 5.
Do you want me to repeat?
Yes, so the question is why iPSCs are better than ESCs, right?
My question was that your notion was iPSCs should replace ESC. Kindly defend that. You've not done that in your speech or your rebuttals.
Team 5, could you guys start answering?
Yeah. So according to the research that we have done, iPSCs, there are diseases like Parkinson's disease in which ESCs cannot be used. ESCs cannot be used because they do not contain the genetic of the cell that we are studying the disease on, whereas ESCs are just pluripotent cells, whereas iPSCs are obtained from the individual. Secondly, there is a major benefit to iPSCs, which is there is no risk of immune rejection, which is seen in ESCs, as ESCs are foreign cells when introduced into a person. When introduced into a person, they can be rejected by the immune system of the individual. However,
Tanya, if I may quickly interject, we all know the science behind it, and I got the point of immunorejection. But the other aspects of it, it increases the risk of a lot of cancers because there is retrovirus which is involved. Yes. Plus, there are a lot of genetic mutations which can happen.
for cancers, there are a lot of vectors that are being studied. Yes, they might not be applied as such, but iPSCs are not applied anyway. Adenovirus vectors in which the genome is not inserted into the genome of the cell. So there is no change in the genome of the cell. Then we have protein vectors. Those do not interfere with the genome of the cell at all. Additionally, we have chemically induced iPSCs and protein induced iPSCs. We have valproic acid, which is an inhibitor of histone deacetylase. It increases the reprogramming efficiency by 100-fold as compared to the traditional method. This mimics the signaling of C-mice. C-mice is the major proto-oncogene that is introduced into iPSC. So if we replace it by valproic acid, we do not need to introduce the proto-oncogene that causes cancer into the cells itself.
Okay, I'm happy you've done your research. Okay, and now my next question for the team, which was speaking against the motion. So basically, there were some concerns about the ethical aspects of embryonic stem cell use, right? Like what we have seen in our clinical practice, the use is practically non-existent, right? So I would like to know from you, what are the present legal aspects? I'm sure you guys would have done the background research about it. What is the present legal status of use of embryonic stem cells? This is my question to the team which was speaking against the motion.
There are a lot of questions and doubts about the ethics and the moral values involved in ESC. And that is why, honestly, it is not in use at all right now, because there hasn't been a lot of clear ways as in how it is helpful, because of the fact that we do not really understand how stem cells exactly work and how, because there's still a lot of research that needs to be done. Secondly,
Sushmita, anything that you can tell me about the Indian scenario that you have read up probably about the Indian scenario? So,
sorry?
That you could have used in your debate probably about the Indian scenario regarding the use of embryonic stem cells. Is it legal? Is there any law which is regulating its use?
Actually, for research purpose, it is very strict and very proper guidelines have to be followed for this. And mostly the embryos which have been used for the research purpose are the embryos from IVF. The selected embryo is being implanted into the for the growth of the child, but the rest of embryos which are being formed, those can be used for the research purposes after proper legalization and all the terms and conditions which have been put forth by the law. And this actually solves the issue of the ethics as well, because either ways the embryos which were being other than the main embryo which was selected, other embryos were going in waste. So other than
also to add, just a second, we could not find a lot of research papers that were based in India on this topic. And I do not really can't recall a lot of penal codes or anything regarding that.
Yeah, so if you could go back and check on the ICMR guidelines, that would be, I think, helpful to you, right? Okay, thanks for your response. Very well researched.
Thank you, ma'am.
Okay, again, as Ritika said, this is something that even I had to also go and read up. It was not something that I was familiar with. Anyhow, so the question to the team 5, that it's a new technology, it is coming up. As already said, can it replace embryonic therapy? So do you see, and you are saying that yes, it should be done. So are you looking at scaling up issues? Can this really be a possible solution for a larger scale use in the Indian context? This is for team 5.
So we could not hear. Could you please repeat?
So my question to you is that it's a new technology, but are we really seriously looking at it because of the scaling up issues? Are we really looking at this as something that will be accessible to larger population? If it stays as a lab, as an exotic thing, which is fine.
Am I audible?
Why are you bothered about it? Yeah, go ahead.
Hello. Am I audible? Yes. So iPSCs are discovered very recently. Only in 2006 were they discovered. And despite the recent discovery, there have been major progress made in the field. There has been research done to reduce the cancer causing the tumor genesis of iPSCs. There has been promising research to increase the efficiency of iPSCs. And therefore, we believe that if so much research in this field is being pursued, then there is a real application because it is used to treat a lot of diseases that the cures are not known of, like Parkinson's disease. Again, we only have treatment of the symptoms of Parkinson's, but we cannot treat the disease from the cause. Again, Alzheimer's, we do not have a known treatment for. So if stem cells, especially iPSCs, are successful in treatment of diseases, then they can be game-changing for a treatment of variety of diseases. So we do believe that they can be used, but research is required.
Okay, thank you. Now the question to the other group. Again, this is trying to argue the other way around. We know technology is difficult, there it is making a lot of errors and the inherent, but tomorrow there could be a much better way of doing the same thing. Getting the pluripotent cells. But as you know, technology can, as a concept, there is nothing wrong. Technology keeps changing and we might have a better technology to do the same thing. So why oppose the use of pluripotent?
Sir, actually, the technology which comes here, always there is a profit and benefit and error cost ratio. So here the cost is the genetic material which is being manipulated or which is being changed gets accumulated throughout the generation, and this might not affect today, but might definitely will affect after certain years, not 80 or 50 years, but it would definitely affect after 300 or 400 years when this gene line gets forwarded more and more. Other thing is also the cost of both ESCs and pluripotent induced pluripotent stem cells almost go equal, because ESCs also require the embryo to be preserved in cryopreservations, and all the necessary steps are to be carried on with it. So the cost is also here the same, and the benefits are also like coming with a lot of cost, like cancer and the gene pool getting disturbed.
Also, to add to this, the fact that we've mentioned a lot of cons of the iPSC right now. I'd like to add that even your point is one of the major cons, that it is not really feasible to the pro that they're saying that we don't have to take it out of any embryo, we can use there won't be immunorejection. All of this also comes at a cost. All of this requires a lot of lab work, a lot of tech work, and it is not easy to make it available and feasible.
And also, if it is easy to get, so there are also chances of misuse as well. So that should also be kept in mind.
Okay, thank you, Dr. Rajadesh.
Thank you, sir.
Yeah, so it's a quite technical topic. The questions are related to the technology and ethics. There, both Dr. Ruchika and Dr. Anand Krishnan. I just want to have a broader aspect. I feel that ESC is established technique. It is therefore quite more than 3 characterize on. So in that case, how you justify this? Because when you say it has to replace, then it will take long time, isn't it? So what is your opinion about that? That once it's a very established this thing, why it has to replace? The technology will modify, technology will improve. So both the technologies will improve. As far as ethical issues concerned, what is your opinion on that?
Am I audible?
Yeah.
So
that is for team number 5. Yeah.
Yes. So we know that embryonic stem cells have been discovered in the 1980s, whereas compared to that, induced pluripotent stem cells have been discovered only in 2006. Despite that, they are gaining popularity. As my worthy opponents said, embryonic stem cells are not being used practically. So the disadvantage of both are no feasibility. But if we work on that, I believe induced pluripotent stem cells
Recording
are superior
stopped.
because they do not have the ethical disadvantages that ESCs propose. In ESCs, you are using an embryo, even though it is discarded. It is unethical to use a incompetent subject without a legal surrogate, and here the incompetent subject is the embryo.
Esteemed organizations such as United Nations also agree with our stance in that an incompetent research subject, their consent cannot be taken without a legal surrogate. And over here, there is no direct therapeutic benefit to the incompetent research subject, which is the embryo in question, because research done on embryonic stem cells is not going to benefit the embryo itself at that point in time.
Okay. So the second team number 8, I think, team number 8. So if that is the argument, what the opposition team is giving, remember the risk of change in the genetic material and risk of the cost also will, as the induced thing will go ahead, the cost will escalate further. And ethical issue is the only thing which is on this side. But what about the chances of developing cancer? Because cancer is something which everybody is extremely scared of. So how will you answer that question? I think this is for team number 8.
So actually, we are against the induced pluripotent stem cells. So that is also, we are saying that cancer can be, just as I said, there are multiple lesions occurring to chromosome number 14, 21, and 22 because of this induced pluripotent stem cell therapy. Moreover, other like Parkinsonism disease, for which this technology was supposed to work, but it has a variable yields at all the times, and the margins of like not getting well is huge. Like the percentage is huge that they can get a desired result. So that is what we are trying to say. Cancer and all the genetic material which is getting disturbed is a major issue for this pluripotent stem cell, induced pluripotent stem cells.
And I'd like to just request the judge, Dr. Geese, to let us know if this question was for team 5 or team 8, because cancer is a problem that is being supported.
Of induced pluripotent stems.
Yes, yes.
Yeah, so
more with induced pluripotents.
We were against the topic. We were against induced pluripotent.
I think there was some mistake. It was for the other team. Sorry. Yeah. I was confused between number 5 and number 8. Yeah.
So one of the major disadvantages of induced pluripotent stem cells, as you have said, is cancer. However, there has been research to improve this. Instead of using retroviral methods, we are using adenoviral methods, which do not incorporate their genome into the host DNA. Therefore, the chromosomes will not have chromosomal anomalies. Another protein transduction method is also used, and reprogramming without these DNA intermediates should eliminate the risk of tumor genesis. In the adenovirus method, no tumor formation was reported according to a research conducted in Stanford. Additionally, we also will introduce well-differentiated cells using genetic markers so that there is no risk of there's no risk of tumor genesis. Additionally, there is a suicide gene which has been discovered, which when introduced to the cell will lead to self-destruction of tumors which are created. This can be supplemented with antioxidant therapy, which shows tumor suppressing properties. And finally, as I said this in my answer before, valproic acid, which is an inhibitor of histone deacetylase, increases reprogramming efficiency by 100-fold, while it mimics the signaling of C-mice. C-mice is the proto-oncogene which is introduced. Therefore, we do not need to introduce the proto-oncogene if we substitute it with valproic acid.
Okay.
Thank you.
For the other team, I think team number 8 now, or team number 5, I'm little getting confused. Team number 8, I think.
8 is against.
Yeah, team number
Yes,
8.
sir. Team
So the
8 will
question
speak
is,
against.
can you hear me? Because I'm not getting the sound at all. Hello?
Yes, sir.
We can hear you.
So my question is the problem of immunosuppression is also a significant problem. Of course, these are technical questions. Most of the questions you answered, but the immunosuppression, which is the problem with ESC. So that is also a significant problem. So what do you want to say about that?
Sir, immunosuppression today is also a problem with other treatments such as transplants and liver transplants, graft, and all. Graft rejection is also one problem, which is today also tackled by using another method such as immunosuppressants and other treatments. So similar with the ESCs also, we can go on with this. Moreover, other than transplants from other body, ESC would be more better because it is from the embryo, and we can get more detailed and more known substance, known organ or whatever we want from the embryos. So the problem which are we already tackling, that would be the same problem, and we would be able to tackle those problems as well, I guess.
Whereas with reprogramming, there are chances that we might discover problems that are not already present, and that would be something that would be more difficult to tackle.
Okay. Okay, thank you very much. Thank you.
Thank you.
Thank you, judges.
Thank you, judges, for this great round. Moving on to the closing statements. Each team gets 1 minute to complete the statement. We start with the proposition team and then the opposition team.
Hello, am I audible?
Yes, you're audible.
So I'm going to start with the closing speech for team 5. Anything you can do, I can do better. That's what IPSCs would say to ESCs. IPSCs, while being morphologically identical, serve as the ethically superior alternative to ESCs, while providing clinical benefits such as tissue regeneration and repair, organ transplantation without the need for immunosuppression, and disease modeling. IPSCs are more accurate with disease remodeling, as they are able to replicate the precise epigenetic and intracellular conditions. This is especially important in diseases like Parkinson's and Alzheimer's, where ESCs fall short. With recent advances, the risk of tumorigenesis has reduced greatly by introducing genes for self-destruction of tumors and antioxidant therapy. With the piggyback system of gene transfer, efficiency of the process is comparable with retroviral methods with added benefits of safety of application. With this plethora of evidence in front of us, wouldn't you say that induced pluripotent stem cells can replace embryonic stem cells in clinical application and disease remodeling? Thank you.
The opposition team can start with their closing.
Thank you. So as we have talked a lot about Parkinson's and how iPSC was one of the major trying cure for it, we've understood, as my partner has said, that it in fact leads to variable progeny with respect to neural and cardiovascular tissues. So it makes no sense to put so many more years of research into iPSC where it is not even fulfilling its own purpose. Now we all know that this is about reprogramming, and that could lead to several misuse, manipulation, and errors. And I'd like to give an example, saying that there was once a person who went for a facelift and was given a stem cell therapy, and then apparently she had bones growing in her face. This is some error that we have no idea about and is very difficult to tackle. Now, there could be regulatory agencies to tackle all of it, but living in India, unfortunately, we know that illegal activities still do occur. And denying the potential of stem cell therapy and iPSC, but we need to remember that we do not really know how to control this. And thank you.
Sushmita. So now that we're
Okay, team 5 and Team 8, we thank you for such a great debate. Now we're towards the end of our event. So on this occasion of Guru Purnima, I would like to take the opportunity to thank all the judges for their valuable insights and constructive criticism. Ruchika ma'am, Anand sir, Rajadhyaksha sir, we really appreciate you taking time off of your busy schedules. It was an honor and a privilege to have you on the panel of the jury today. I would also like to thank all the participants for their enthusiastic participation. You all were amazing today. The results will be declared on 15th of July. So before we conclude, I would like to ask the judges if they wish to share their thoughts.
This is Dr. Anand. I think, first of all, let me thank the organizers for giving me this opportunity. I was very happy that these kind of debates are happening as a part of festivals, and not just, let's me put it in a way, more frivolous activities. These are more academic, and I think these are important to develop the personalities of the future doctors. And I'm very happy to be part of this. And I think overall the quality of debate was very good. Even if somebody will win and somebody lose, but I can at least say from my marks that the gradient is not very big, that somebody should be very worried that they did not do well and others did very well. So you all the best. Thank you.
It was a pleasure having you.
Am I audible?
Yes, ma'am, audible.
Yeah, I would like to congratulate the organizers. It was a very well thought out debate, and it's taken debating to a whole new level with the rebuttal and the kind of intricacies that were involved. And the topics also were very well thought out. They were very contemporary, and I mean, a lot of them required even me to open up my books and read about IPSCs and ESCs. So, and the topics were very, very well thought of. Kudos for that. And lastly, the participants were all amazing. I mean, the kind of maturity and the kind of research-oriented thinking that you guys have at this young age, it's commendable. And I love being a part of it. So thank you so much for inviting me.
Thank you so much, ma'am. We really appreciate you taking your time out and being with us today.
Okay, so thank you again. Thank you, Shruti. Thank you, Shailaja. Thank you, all your mentors. GS is always means quality, and you have maintained that particular thing in this particular debate competition. The participation was good, and the topic selection was excellent. I think it is so reassuring to see that such a bright young, budding doctors and how they debate so fluently with such a thorough research. And I hope they remain in India for their practice, and they are not stolen by the Western world from India. But with that hope, I think it was excellent debate, and thank you very much for involving me, and all the best to all of you. Thank you.
Thank you so much, sir. I know the Mumbai rains and the busy schedules, but you could make it. I'm really grateful to you today. Thank you so much. Okay, so we would conclude this session today. Thank you all the judges, all the participants. Thank you so much. Take care. Meet you in the final round. Yeah.
Thank you. Bye-bye.
Bye-bye.